Should I start sharing my screen so that I cannot share screens, disable.
You have to be enabled here.
Yeah, you should be able to now.
Yeah, is it visible?
Yeah. Can you also change once and the slides? Yeah, perfect. Working. Maybe wait for 1 or 2 more minutes.
Yeah, sure.
Maybe it's the Dasara effect.
Yeah, you might get some background. There is some house is quite close to place where there is a lot of festival. So there could be some disturbance coming in once in a while, announcements and stuff like that. So please excuse.
Maybe we should start now, Soumya, or we can wait for some more. It's 9:31.
Yeah. Okay, let's start then, or? Okay. Okay, so good morning, everyone, and welcome to the day 2 of Soft Marriage and Investigative Meeting. Happy Dashera to all of you. Then now we will begin the first session of today, which is on biological soft matter. And I, along with Dr. Balakrishnan, would be chairing this session. So I'll introduce him and then he will take on this task. Srinath Balakrishnan is currently at IIT Bombay. He studied mechanical engineering from National Institute of Technology, Calicut. Then he did his master's in the same in Virginia Tech, followed by PhD in Biosystem Science and Engineering from ISC, where he continued doing postdoc, and now he works in areas of mechanical modeling of biological cells, phenomena, biomechanics, so on and so forth. So without much ado, we look forward to hearing from you, Srinath. And just one point that when you have, when anybody wants to ask questions, you can please raise your hand, unmute yourself and ask. Otherwise, you can also put it in the chat box. So please, Srinath, the stage is all yours. Thank you.
Okay. Yeah. Okay. Thank you, Dr. Shobana, and thanks for inviting me and also asking to be there for the session. Yeah, so I'll just go ahead with my presentation. So my work has been in my PhD and continuing afterwards is on looking at the shape of the nuclei. And so this is artwork that I made as part of the first publication that came out of that work. And here I just want to take your attention to a couple of aspects. And so this is more like an artwork. It just tells a story in a very kind of slightly abstract way. So you can see that there is this green colored surface. It's like a surface, like a three-dimensional surface. And what you see here in blue are shapes of nuclei that has been obtained after doing confocal imaging and a little bit of image process. So you see that these nuclei are actually embedded on the surface. They are on the surface, and they are not all over the surface. They are actually in, like, it's like a line it looks like a highway, and these nuclei are all studded there like pebbles. So we will see in this presentation why this picture is like a decent abstraction of the kind of work we are talking about. So first, just a brief introduction about nuclear morphology, the shape of nuclei inside cells. It has been known for a very long time that it is an important kind of a biosensor. It integrates a lot of signals and it gives something like an output. So it is known that nuclear shape is altered in diseases, and the very famous pap smear test, one of the important points is to look at how the shape of the nuclei are different. So in this one, typical nuclei are supposed to be of this size, and then you have some enlarged nuclei, which tells you that something is wrong. And this is a very famous test for, I think, for cervical cancer. Also, it is also known in this accelerated aging disease, which falls under the class of laminopathy, which basically is a mutation in a gene which makes this meshwork inside the nucleus called lamin. And once that meshwork is broken because of some genetic mutation, then your nucleus is actually misshaped, and that is a very important marker of the disease. And there are a few other things like in senescence, aging, and many of these things we know that the nuclear envelope is altered, and that actually changes the nuclear shape. So these are some of the very drastic ideas in the case of diseases, but also there have been a lot of studies which looked at perturbing different mechanical elements in the cell. That is the most obvious one is a cytoskeleton. You have their actin, microtubules, and the specific protein lamin, which is an intermediate filament. If you perturb any of these mechanical elements in the cell, then it can change the shape of the nucleus. So this has been very well characterized. Also, there is a osmotic pressure imbalance between the inside of the cell, that is the cytoplasm, and the exterior of the cell, as well as there is an osmotic pressure imbalance between the nucleoplasm and the cytoplasm. So these when you change the osmolarity in the outside the cell, you see a large changes in the shape of the cell as well as the nucleus. So this has been very well characterized. Also, if you change the chromatin, like if you kind of disassemble the entire chromatin using some drugs, you see there are some drastic changes in the shape of the nucleus. So it is known that nuclear shape is actually regulated by many of these factors. But the kind of question that we try to ask is that, okay, so now can you look at the change in the nucleus shape and then say what change caused it? So something like an inverse of that. So we are not looking at changing something to the cell and then looking at what changes it caused to the nucleus. We are trying to say the reverse question, where you look at the change in the shape of the nucleus and then can we say something about why that specific change happened. So one of the things I just wanted, just a simple example of how such a thing can be done. Like a very simple example is if you have a cantilever beam and then if you had a moment at the free end, then if you use a very famous Euler-Bernoulli theory, you see that the curvature is given by m by ei. So this m is the moment, e is the elastic modulus of this beam, and I is a geometric parameter, which is the moment of inertia of the cross section. So now let us say you observed two beams, and each of them have the same boundary conditions, meaning it is fixed at one end and you have a moment at the other. And let's say we know that this is the situation. Let's say we know that these two beams have the same forces, or I mean, the same boundary conditions. Then if by just looking at the geometry, so this has more curvature than the other. So the curvature of this beam is more than the other. So just by measuring the curvature, you can actually say something about this ratio. So you can say the ratio of the moment to Young's modulus times the moment of inertia of the cross section is less in case 1 to case 2. Now, this is actually going from the shape to actually mechanics, because these are mechanical properties. This is a force and these are the mechanical properties of the system. This is a material property and this is a geometric property. So just by looking at the final shape, and if you know the mechanics of the system, you can actually talk about something more than just the shape. You can talk about actually the forces, the properties, and everything. So we use a similar kind of idea to actually do the same analysis with the case of a nucleus. The mechanics will be slightly complicated, but the idea will be the same. So let's look at, so cell is obviously a very complicated system. You have 10,000s of genes, same number of proteins and lipids and all sorts of other things. So such a complicated picture is not amenable to any kind of mechanical analysis. So we need a much simpler version. I mean, a simpler mechanics picture, which actually essentially captures the essential features of the mechanics. So let us say we take a cell, and here I am representing only the some of the things. So this red is the actin cortex, which is known to have a tensile force. It's known to be under tension. And here you have the blue is the nucleus, and then we're just representing the chromosomes inside. And if you look at it very carefully, there is a dark shading inside the nucleus, which is basically telling that inside is slightly more dense than the outside. Like the nucleoplasm is denser than the cytoplasm, because of which there is known to be an osmotic pressure, just pushing the nucleus outward. Is also representing some microtubules. So this is itself a very slightly complicated picture, but one of the very famous theories about cell mechanics is the tensegrity theory, in which Professor Donald Ingber, who is now at Harvard, he founded the Wyss Institute. He showed that tensegrity cells, the cells act like a tensegrity system, which means that the stability in the cell is achieved through tension. And he proposed that actin is the tensile element, which is under tension, and microtubule is the compressive element. So so that is a more microscopic picture where you have the actin and microtubule. Let's say we look at the cell as a whole, then we can actually think of it like a tent. So this analogy is is also presented by Professor Shamik Sen in his mechanobiology lectures, which are there on YouTube. So we can think of the cell as a tent, wherein the actin cortex is like the canopy, which is under tension. And so whenever you have a tent, you have a tensile element, which is the canopy. And then to balance out that tension, to prop it up to a height, you need a compressive element, which is a pole, typically when we use tens. Now, in the case of a cell, we can think of this compressive element as the nucleus, which itself is an inflated spherical membrane. So because of this the osmotic pressure imbalance between the inside and outside of the nucleus, you have an osmotic pressure, which is actually inflating it. So the inflated membrane acts like a compression element, which is propping up this tensile element, which is the actin cortex. So this has there are so many experimental observations which backs this mechanical picture. Another interesting, I could say more of an intuitive argument is that a tent is a structure which can give you stability with minimal material. That is why we actually carry the tent for treks or something, because the only heavy element is the pole, and the rest of it is like a cloth, right? So if we were to think of an optimal structure for stability, optimal in the sense of minimum material use, one would actually think of a tent. And since evolution is an optimization procedure, so you would think that life has actually come up with this solution for designing a cell. Okay? So this analogy helps you to think about how cells are. Now, in the specific case of the nucleus, what you can do is we can actually simplify this picture even further. So this picture is like an inflated balloon. So if you have a balloon, you inflate it, and you take a membrane, you stretch it, and then you put it on top of the nucleus. So on top of this balloon, that can kind of describe how a cell is. But this can be simplified further, because typically this dimension of the cell is much larger than that of the nucleus. So if this dimension is much larger, then this surface is where this cortical actin is contacting the nucleus is almost flat. So since it is almost flat, you can replace it with a flat plate. So so our original picture was like an inflated balloon, which is pushed down by a stretched membrane. Now we are replacing the stretched membrane by a flat plate. And the reason is because the cell is much larger, so this region can be flatter. Is very flat, so you can assume it to be exactly flat. So now this picture is actually now very simple. So we have a membrane which is inflated by a pressure P and which is pushed between two plates using a force F. So now this is a simplified mechanical picture which can be used for a mechanical analysis. So this analysis was developed in 1973 by a group from, I think, Carnegie Mellon. So they looked at the exact same problem. So they were looking at it in the context of tires, where you have, let's say, a membrane which was spherical with an undeformed radius capital R, and that has been inflated by a pressure P, and which has been pressed between two flat plates using by a force F. So now, if you were to do the mechanics of this one, you get these equation, one for the tangential direction of the membrane and one in the perpendicular direction. So this equation in the normal direction is pretty famous. This is the same soap bubble equation where you get P equal to 2 sigma by R. That is a special case when the tensions in both directions are same. It's T1, T2, so two orthogonal tensions. And if when T1 equal to T2 equal to sigma, and if the radius of curvature is also same in the both directions, which is like a sphere in the soap bubble sphere, then you get the P equal to 2 sigma by R. That is the famous soap bubble equation. And this is the general equation in the case when tensions are not equal. So if you were to apply this equations of equilibrium to this system, and if you assume Mooney-Rivlin material model, you can get these relations for tensions and curvatures. And if you plug these tensions and curvature back into this original equation, what you will get is actually you get these set of non-dimensional parameters. So if you, this is just a substitution to the second equation, that is the equilibrium in the direction normal to the membrane. You see that on the left hand side, you have deformation parameters. These are your strains, which basically tells you what is a deformed configuration of your membrane. And on the right hand side, you get a nice non-dimensional number. So this, I call it eta1. That is the pressure by the elastic modulus. And it is also scaled by the geometric parameters. R is the undeformed radius of the spherical membrane, and H is the thickness. So this you get your first non-dimensional parameter. And you also get a second non-dimensional parameter, that is by looking at the force balance in the vertical direction, that the force F is the vertical force that is the plate is pushing down on the membrane, and P, the pressure is actually resisting that force. So if you were to use that force equilibrium equation, then you get another non-dimensional parameter eta2, which is actually force divided by P, square root of that, and is also scaled by the radius in the undeformed configuration, that is pi r square. Now, these are not the only non-dimensional parameters you can have. So, okay, so this system is governed only by two independent non-dimensional parameters, but there can be other sets as well. So these are the non-dimensional parameters that I discussed before. This eta1 is this ratio, pressure by Young's modulus, and eta2 is the ratio, is the square root of the ratio of force by pressure. And there can be other non-dimensional parameters which are not independent. So once you specify eta1 and eta2, the mechanics is fixed, and then you can calculate these parameters lambda 0 and 2. Lambda 0 and 2 themselves are independent, but they are not independent of this set and this set, because there are only two independent parameters. Now, what is lambda 0? Lambda 0 is the stretch at the apex, that is the deformed length divided by the undeformed length at that point. So that is the elastic stretch at the apex point. And just to say, eta2 is actually a measure of the area of the flat region. So eta2 is the radius. This is the radius normalized. So this is the radius of the flat region divided by the radius of the undeformed sphere. So that is eta2. Now, if you take this flat portion and if you map it to the undeformed configuration, so this is the flat portion in the deformed state. In the undeformed state, the flat portion becomes a curve, and now this flat portion can be characterized by an angle tau. So now this angle is also a measure of this flat region. So that is the how much of the nucleus has become flat. So that is this angle tau. So we can characterize these mechanics using two non-dimensional parameters, eta 1, eta 2, as well as lambda 0 and tau. These are not mutually independent, but among them they are independent. Lambda 0 and tau is independent, eta 1 and eta 2 are independent. Now, since you have only two non-dimensional parameters, if you take any three shape parameters of the nucleus, there should be some relation. So let us say I take some three shape parameters. Let's say I take the volume, I take the surface area, and I take the projected area. Projected area means if I look at this shape from the top, I will see a circle. Now, if I take the area of that circle, that is another measure. So you can take any three shape parameters. So in this work, we have taken three shape parameters. That is the projected area. That is, if you look from the top, the area of the circle, the volume of this shape, and the surface area. So if we take any three, there should be some relationship between them, because fundamentally it is the mechanics is dependent on only two non-dimensional parameters. So so that relationship is given by this surface. So if you were to draw this space where you have projected area along the, let's say, this axis, and then surface area along this axis and volume along this axis, there is a relationship among these, and that is given by the surface. So what the surface means is that if any object behaves as if it behaves according to the mechanics determined by this problem, this mechanics problem, then the final shape parameters, that is the projected area, surface area, and volume, should fall on this surface. Or conversely, if you have shapes which fall on that surface, they should probably behave according to the mechanics given by this problem. Okay? And then on the surface, we have plotted the contour lines of these non-dimensional parameters. That is, eta 1 and eta 2 behaves slightly, they are, I would say, they're not really orthogonal. They have an angle. These are the contour lines. And if you see lambda 0 and tau are actually.
We have a couple of more minutes. Sorry.
Oh,
yeah.
Oh, that's it. Okay. Oh, sorry. I think I'm not even close. Okay, fine. So sorry. I think I did not realize that. Yeah. So what we did was we looked at the shapes of these nuclei, and we had an image processing algorithm to measure the shape of the nuclei. And we saw many different nuclei. And what we found out, first we did this study with hepatitis C virus in liver cells, and we found out that actually individual nuclei from these normal liver and hepatitis fall on the surface, saying that our mechanics guess was actually quite close. And we made a prediction by looking at these non-dimensional parameters, how these non-dimensional parameters changed. That the non-dimensional parameter was lower for our normal cells to compare to our HCV cells. And because of that, we predicted that the elastic modulus might have decreased. And that we verified using experimental studies, that we found out that this lamin protein is actually downregulated, as well as the nucleus stiffness is actually lower. And then we also applied this model to multiple cell types, different kinds of cell types, after that first study was over. And we found out that all the cell lines actually fall on that surface. So we have these 5 cell lines we have measured hundreds of nuclei in each of them, and all of them fall on that surface, telling that in general nuclei probably behave like the mechanics that we have described. And not only that they fall on the surface, they actually fall along a straight line on the surface. They are not like, if you take one cell line, they don't fall all over the surface. They are falling along the straight line on the surface. And this straight line is along this direction, lambda 0. And this so this is actually the principal variability. And then you have another principal variability, which is the perpendicular. So the cells fall along the principal variable direction, that is along lambda 0. So that told us that these parameters form a canonical basis for characterizing the nuclear shape. And the variability in nuclei in a cell population is along this lambda 0. We did some other simulations, and also we found out what is the geometric interpretation of these parameters. So this lambda 0 is like an isometric scaling. So that is like you're taking the nucleus and they are scaling it. And this parameter tau is actually the flatness, how flat it is. So what that means is that in a cell population, the nuclei vary because of scaling rather than flatness. The flatness is approximately same. The scaling is what is different. Okay, and then we did some biochemical experiments by depolymerizing different parts of the cell, like actin, microtubule, and by growing cells on different substrates. And we found out that this parameter tau actually correlates with actin tension. So the more tension, the nucleus is flattened more, and that actually increases tau. So tau actually correlates with actin tension. And to summarize this work, this is the last slide. So we say that the nuclear shape can be described by these two non-dimensional parameters. They form a canonical basis. And this parameter tau indicates flatness, and this lambda 0 indicates the scaling. And tau correlates with actin tension, and lambda 0 anti-correlates with elastic modulus. And our main idea here is that if you put physical modeling into your experimental observation, you can probably add some more insights. So in this case, your experimental observation is the nucleus shape. Your physical modeling is your mechanics, and that tells you something about your actin tension and the elastic modulus of the envelope by just from the analyzing the shape. Okay, so sorry, I think I had to rush the last few. So yeah, I'd like to thank my advisors and the BSSC and funding sources. Yeah, thank you. Sorry.
Thank you, Sheena. Thank you for this exciting talk. We do have a couple of more minutes for questions. Do we have any questions? Okay. Yes, Vibhu, please.
Hi, Sheena. An interesting
Yeah.
talk. I have a couple of questions regarding this. So when you said all like in a particular for a particular cell line, they fall on the lambda 0 line, right?
Yes.
And that's not accounting for the fact that cells are perturbed outside. For example, you compress them or you kind of mechanically perturb them. So even if you do that, will they still follow this lambda 0 line?
So okay, so we haven't done any kind of stretching experiments or anything, but the kind of perturbations that we have done. So we have depolymerized actin filaments using the cytokalasin B. We have depolymerized microtubules using nocodazole, and we have grown these cells on different substrates. We have not done any active measurements till now. I'll tell you something that we're going to do. In all these cases, we see that they're still falling on the straight. So we have done, so okay, so I would say that we have just growing the cell lines, 5 of them, and perturbing 3 perturbations. And even though I've shown only one cell line, we have done these perturbations to all these cell lines. And then in every case, they fall on a straight line, and the straight line that it falls on shifts according to the perturbation that you have done.
If I may continue just in the same line, so when you cells has to go through a confined geometry, I mean it itself kind of tunes its nuclear shape, according to the geometry. So will that also follow in the will that also follow in this parameterized space? And just take continuous, yes.
Yeah, I feel that it will fall on the parameterized space, but I'm not sure whether it will fall on a single straight line anymore.
Okay.
So for example, at least I know that when the cells have to divide, they actually kind of almost detach from the surface, right? So at that time, they would actually move from here to a lower angle of tau. That is like tau 0 means spherical, so they should move from let's say this line to that line. Yeah, they should definitely move, but I still believe that they will fall on this surface.
Okay. Yeah. Thanks a lot.
Yeah.
Okay. Can we have Siva now to ask his question? Yeah.
Okay, so I have a rather naive question. Excellent talk, by the way. The question is, like, when I think about cells, I always think about it as viscoelastic system, right?
Okay.
And I'm surprised that all your model just says that it is an elastic system, and then it beautifully fits in. So what would you comment?
Yeah, so what you said is perfectly right. So I think it's about the time scale. So we are looking at the shape of the cell. We are looking at we are looking at the shape of a cell after it has spread out. So whatever viscoelastic effect that might have been there should have evened out by then. So it would have, slowly it would have come to its resting shape, because it's a very slow process. Spreading of cells usually takes around 5 to 8 hours. It's a very slow process. So at the end of it, it has reached the position where it is actually now probably elasticity actually works. because the rate of change is actually very small.
So it's basically not that like you are at long times for viscoelastic system, I would rather expect the viscous effects to take over. But what I understand now is that like you're rather getting adsorbed to the surface that you are looking at, and hence you are not letting the system to take over with the viscous effects. Is that how I see? Because if this is
that this might be a viscoelastic solid rather than a liquid. So there is difference. Like solids,
I agree.
Yeah.
Could be, but usually people did not found shear thickening behavior for cells. Is that right?
That I don't know.
No, yeah, I'm also not very.
Yeah, that's what. So I was thinking these to be shear thinning systems. So in that sense, if you just wait for such long times, I would rather expect it to be a simple like a Newtonian liquid which is flowing, right? So even then, if you're if you can measure your tau to be like rather constant, and like it just like reached some steady state, so then it means that something else is constraining the cell to be there. So I don't know what is that.
So okay. So at least we have looked at the nucleus. I'm not very expert at fluid mechanics. I'll tell you right away. But still, so what we looked at is that we looked at. It's not very clear from this picture, but okay. So actually, what we have plotted for this nuclei is actually the curvature. If it were, let's say the nucleus, I'm talking specifically about the nucleus. If it were a fluid and then dominated by like surface tension, then wouldn't you expect the curvature to be constant on the surface?
Yes, that's
Yeah.
right. I mean, you cannot expect this kind of sharp edges that you're drawing.
Yeah. So actually, we see that this is not. I mean, so this is actual real shape of the nucleus. So this is actually these are confocal images of the nucleus, which I run some image processing algorithm I did, and then you get the shape of the nucleus. So when you look at the shape of the nucleus, you actually see very large changes in curvature. So I don't think we can think of it like a liquid.
Sure. Thanks. I think like I'm taking more time than what is needed, so we can catch up maybe in the discussion.
Sure.
Yes. I have Nitin now to ask.
Yeah,
last
I'll just ask a quick question. So, so Srinath, nice talk. But so does your study also answer in some way why nuclei always lie in the center of the cell?
No, we haven't looked at the nuclear positioning yet.
Because like when you give the tent analogy, that would naturally place the nucleus in the middle, right?
Yes.
Because I don't know whether there is any mechanical understanding of this thing, like why nuclei are always seen in the middle rather than at the edge.
Not even true, right? I mean, you don't have the nuclei always at the center.
I mean, when cells are moving, they might move somewhere else, but I don't know.
So
yeah, there are a lot of work.
Yeah, exactly.
There is
Please
a lot
go
of
ahead.
work on nuclear positioning. There is actually a lot of work, like they acts like piston, yeah, there is a lot of I would say very molecular details. Like if you perturb this, then this does not happen. When you perturb that, then does not happen. But a very mechanistic understanding, like from a mechanics perspective, I'm not very clear from reading the literature. So as I think both of you are correct in the sense that for many kind of cells like epithelial cells, the nuclei is bang in the center. But there are many other kind of cells wherein nuclei are not really in the center. It is somewhere, sometimes in the front, the leading edge, the back edge, and everything. So it's actually a very complicated picture.
Okay, thank you.
Thank you. Surabhi, may I request you to please ask your question later, during the interactive session so that we can start the next speaker. Would that be fine, Surabhi?
Yeah, that's fine. Thanks.
Thank you. Okay, thank you, Srinath, for this wonderful talk. Just so that we are on time, I would request now Srinath to chair the remaining part of the session and introduce the next speaker, Dr. Medhavi Vishkarma.
Yeah. Okay. Thanks, Shobna. Hi, Medhavi. Welcome to your talk. I mean,
Hi.
we are happy to have you here.
Hi, everyone. Thank you. Thank you for having me here.
So, Medhavi obtained her BTech in biotechnology from Punjab University and went on to do an MTech in biotechnology from IIT Kharagpur, followed by a PhD in bioengineering from EPFL Switzerland, and a couple of postdocs from Max Planck and University of Bristol. And now she is an assistant professor at the Department of Biosystem Science and Engineering, which is my alma mater at ISC. Very warm welcome to the Soft Matter Young Investigators meeting, and we can start your presentation.
Thank you, Srinath, for the nice introduction. I'm just gonna try to share my screen. So, okay, how do we share screen on Zoom?
There's a share screen button, right?
Ah, yes, I found it, because we use Teams here all the time. It's a bit... Share. Okay. So everybody can see my screen?
Yes, yes.
Okay, great. So welcome everyone, and thank you for being here on Dussehra Day. Happy Dussehra, by the way. So actually, I'm gonna, because the audience is soft matter related, so I'm going to present two of my research stories, which are more related to soft matter, and I'm going to try to explain or try to demonstrate how we can use soft matter physics to understand concepts of wound healing in epithelial biology. So the title of my talk is Dynamic Heterogeneity Dictate Leader Follow Dynamics During Epithelial Wound Closure. So I'll be talking more about wound healing and the concept of dynamic heterogeneity, which is a very well-known concept in soft matter physics. And we'll combine them together to see how in epithelial cells that actually can resemble a lot like soft matter, how this dynamic heterogeneity play a very important role in wound closure. So let's go to the next slide. So epithelial tissues, for the benefit of people who are not from biology, just very briefly, they are the cells or tissues are actually lining almost all the organs of our body. And when they are lining our organs of our body, they form a two-dimensional monolayer of cells. So the monolayer of cells look like this. And when you look at them on a longer time scale, the monolayer is not very passive, so it's very active, it's very dynamic, because there's a lot going on in the monolayer. The cells are trying to maintain a specific density at homeostasis, and they are trying to regulate that density by removing cells, and so it's basically cell division and cell death that's regulating the cell density at homeostasis. So that dynamics that you see, that's coming from cell division and cell death. And this dynamics is actually very important, playing a very important role in maintaining this dynamic heterogeneity that we'll talk about in the next few slides. How epithelial tissues resemble soft matter. So as you all might already know, that it's a very analogy is very it's a naive analogy for a very not for this audience, but I'll just go ahead still. So when you have diffused particles that are at a very low density in a fluid, you see that they are when the particle density is low, they are able to diffuse very freely and move about and exchange with their neighbors. But as you increase the density, so this is a very characteristic of all the soft matter, non-equilibrium matter like colloidal particles, sand, grains, foam, all of the matter that actually solidify in a disordered manner when you cool them. Like for example, a supercooled fluid, when it transitions from supercooled fluid to a glass, it will convert into it will exhibit characteristic like this. So meaning when you increase the density of a particle or you decrease the temperature, you will obtain a coordination, or you increase the cooperativity, because in order for the particle to exchange their neighbors, they will require a cooperative movement. So they cannot really move independently, but they have to cooperate or coordinate their motion with their neighbors. And this motion coordination is increasing as the density is increasing. At the same time, there is a kinetic arrest that is a characteristic of dynamic heterogeneity, which is coming up when the particle density is increasing. So similar characteristics you can see in epithelial tissues, that when there is density increasing, when you go from low density to high density of epithelial cells, you obtain an increase in coordination of movement, and you obtain a kinetic arrest. So cells are being arrested, so there's less movement, but there's whatever movement is there, that is all coordinated or correlated. So there is increased coordination between the neighbors and there is less movement altogether. So this characteristic is very similar to non-equilibrium matter, that you see in nature, and you can also appreciate it with this graph where you see that the number of cells that coordinate increase when the cell density is increasing, and there's decreased movement that you can see from here. The velocity is decreasing when the cell density is increasing. So when you put this together in the jamming phase diagram, you will see that you can actually make a similar jamming phase diagram that you have seen, must have seen in other non-equilibrium matter like glass or colloidal particles or grains, jammed grains. So in this case, the parameters that control jamming and unjamming is temperature, density, and load. So basically, when you decrease the temperature, you have a jam state. When you increase the density, you have a jam state, and when you increase the load, you have an unjam state. Similarly, in the cells, when you have cell adhesions is decreased. So basically, when there is lower adhesions, there will be unjamming, and then when there is more adhesion, there will be jamming. When there is more motion, there will be more unjamming, and when there is less motion, there will be jamming. And as similarly, you obtain a hybrid state where there is jamming and jamming coming together. So it's more of a dynamic heterogeneity state where you see cluster of cells coming together. So the cells that have more motion come together and be together. So this cooperation you see, and this hybrid state is what we actually see in epithelial cells more often. And this actually, you can appreciate this dynamic heterogeneity or this jamming and jamming behavior in epithelial tissues by doing this traction force microscopy and obtaining this stress profile of cells. So you can see peaks of high and low stresses where you can see that the cells are coordinating their forces with their neighbors. So the question that we asked is actually we were actually motivated by this paper that I'm just going to show you very quickly. So this was a paper that came up in 2018 and showed that actually there is a physiological relevance towards this jamming and unjamming behavior that you see in epithelia. So what they have done is they have taken cells from asthmatic patients and cells from non-asthmatic patients and have shown that the patients with asthma have cells, epithelial cells that are more unjammed. So cells are not behaving as, so cells are not getting jammed, and their mechanical behavior and tissue basically jamming is different as compared to cells that are normal and does not have asthma. But the biological relevance is not very clear still why the unjamming would be required in asthmatic patients, but there is a certain extent of unjamming. So you can appreciate it with this cell shape index, that the cells are more, the shape index is, so this is perimeter by square root of area, this parameter, and you can see that this is much greater for asthmatic patients. And here you can see over time the jamming is not happening. So you can see that there is a decrease in cell shape, but that there is an unjamming that's characteristic of asthmatic patients. While when you don't have asthma and you increase the density, you will go from unjamming to a jamming behavior. So what we asked is how can we if we can find some relevance of this jamming behavior on wound healing. Basically, if you this characteristic behavior of epithelia that you see peaks and bases of high and low stresses, and this unjamming and jamming, this dynamic heterogeneity inside the monolayer that you see in the epithelia, does it really can it really affect wound healing, or what is the relevance of jamming behavior on wound healing? So it's what we see. So what we are doing is what we did is actually in this study is just grown epithelial cells on a two-dimensional dish glass cover slip, and then we did a wound on the cells, and cells saw the wound and cells started to migrate over the wound and try to fill the gap quickly as possible. When you have this similar system in actin stained cell line, so this is a cell line that is stained, not stained, sorry, actin is labeled by the fluorophore. So you can see live labeling of actin, and while you image the phase contrast cells, you can also image them in a fluorescence mode and see the actin as you go. So in this case, you can appreciate certain cells that are forming protrusions in the front. So these cells in the front, I don't know if you can see these cells that are actually forming protrusions and behaving as leader cells. So what we asked is whether this unjamming jamming behavior can affect the formation of leaders, because at that time it was not clear how these leader cells are actually forming. So from the uniform monolayer, there's suddenly a wound and then the cells migrate and some cells become leaders, but it was not clear How do the cells become leaders? Which cells become leaders? And what is the regulation? So basically, if this cell is becoming leader, the neighbors is not generally becoming a leader. So there's a leader-follower behavior that we see, but we were not sure, and it was not known how this is controlling and how this is regulated in epithelial wound healing. So we wondered if the jamming behavior, if the dynamic heterogeneity inside the monolayer actually affect this leader-follower dynamics when the cells are suddenly exposed to a wound. So this study is already published and actually some years ago. So I will not go in very much detail of this study, but I'll talk about it briefly, and then we'll switch to a more newer study that we did following up this study. So what we did is we measured forces inside the cells, because monolayer stress is one of the characteristic parameter that you can actually measure and look for dynamic heterogeneity inside the epithelial cells. So how we measured forces is by doing traction force microscopy. What we did is we grow cells on polyacrylamide gels, and this polyacrylamide gel have fluorescent beads. So when the cells move on this gel, these red things are beads, and the cells are moving on the gel. You can actually simply by calculating the deformation of the beads on the gel, you can calculate the force that the cell exerts on the gel. So this is a traction force microscopy that we did. And from the force balance algorithm, then you can actually calculate how much force the cell is exerting on their neighbors, because the forces on the substrate will be balanced by the forces between the cells. So you can calculate the monolayer stress. And we did that. And what we saw very interestingly was that before the leader cells actually form, before if there is a wound here, and you see increased stress in the cells in specific regions. And then what we saw was from these regions, you see emergence of leader cells. So when you go in this movie forward, you will see that there is a leader cell emerging from this high stress regions. So that's showing that there is a high tension that's aiding the leader cell formation. That is what it's representing, or is indicating that there's high tension regions inside the monolayer, and these regions are actually aiding in the formation of leader cells, because you see leader cell forming here, but not here, for example. Or I mean, there's more movies where I have also shown that from the regions where there are no stress, you don't see leader cells, but I'm not showing it here. You can please refer to the paper that's written on the bottom. But when you see go over time, you can see that leader cells are emerging from high tension regions. So what we hypothesize is that cells in the back are actually exerting some kind of force on the leader cells. And since forces is within the epithelia, people have shown that it's mainly tensile. So there's a compressive force element is very low. So there's a lot of tensile forces going on in the epithelial tissues. So what we wondered is if it's all tensile forces, so it could be that these cells are actually pulling onto the cells at the edge and then forming or aiding in the formation of leader cells. So then what we did is a mechanical pulling experiment, because we wanted to see, this was not the slide I wanted to show you, but so you can see that the regions of high stress is forming the leader cells. But what we hypothesized, as I was just saying, that we thought that there's a mechanical pulling going on from the follower cells to the leader cells. So what we did is a mechanical pulling experiment. What we tried to do is try to pull the cells from the back and see if they actually protrude in the front. How we did that is we grow cells on a PDMS substrate, and we made a trench here, and then we let the cells move on this substrate. When the cells crossed this trench, there will be leader cells here and there, but when we will found regions where there were no leader cells, like here. And then what we did is we put this PDMS membrane onto a mechanical stretcher, and then we stretched from the back. So when we stretch from the back, because there is a trench here, these yellow cells will be able to pull on the pink cells, and then you will see like if you stretch from here, the yellow cell will pull on the pink cell. And what we expect is this pink cell should protrude forward, so it should become like a leader cell, because we are putting force in the back is what we had, because this is what we hypothesized. So it's exactly what we saw. So this was the experiment, the real experiment that I'm just showing you now. There's a trench here, and when we pulled from the back, you see these cells are protruding forward. But what also happens is that they retract also very soon. So because there is already an existing leader, so that is a different problem, that there is an existing leader. So that leader is actually inhibiting the formation of other leaders in the neighborhood. Therefore, these cells retract. But in that instance, when we pulled, these cells are protruding forward. So in the movie, you can see, appreciate it more clearly. You can see that when there's a pulling force, you can see that there's protrusion of cells in these cells are protruding, and then they retract because there is existing leaders already present. So this demonstrates that pulling forces from the bulk actually allow protrusion formation in the future leaders. But what we were puzzled by is that this retraction of the protrusion. So that we thought is happening because there is an existing leader, and that leader has a territory of guidance. So that has a territory that it's my territory. Nobody can be a leader in this region now. So this is actually determined by forces again. So what we saw is that these force stress peaks and bases of high and low stress that you see has a correlation length that decays over time, over distance. So correlation function decays about 200 microns, and that means that the cells are correlating their forces up to this region, up to this length scale. The length scale of force correlation is about 200 microns. And what we saw when we did the actin staining over long space in the cells, you see that the distance between the leader cells is very similar to what is the length scale of force correlation inside the monolayer. So the leader to leader distance is very similar to the force correlation length, also representing that the length up to which the cells are able to correlate forces is also the length scale of leader cell formation. So that means the leader cell is also exerting a force up to the similar length scale and pulling onto the followers so the followers can move. So from here, we did some other simulation, like collaborated with some theoreticians and did some simulations on this and tried to actually show that this is actually the case. But I'm not going to present that today. I'll just switch now to a different topic.
Mehavi?
Yes, yes.
Yeah, I think can you wrap up in a few minutes? I mean, I have some questions.
Yes, yes. Let's see how much time. Oh, yes, yes. We don't have so much time. I'll wrap up. I just have a few more slides to show. Just the new work that we did on following up this work. So the dynamic heterogeneity influenced the leader-follower behavior at the wound edge is what we saw. But the question, because the dynamic heterogeneity is influenced by cell density. So when you increase density, there is the cells are packing more, as I showed in the beginning of the first slide, that the cell density is actually influencing the dynamic heterogeneity of all the non-equilibrium matter. So what we hypothesized is, or what we wondered actually, is that does epithelial maintain a certain cell density at homeostasis in order to, from the perspective of future wound? For example, if the density is much higher or much lower, you don't see this kind of a behavior of dynamic heterogeneity, and then that would that affect a future wound? If there is a wound, the cells would not be able to efficiently migrate and so on. So that is what we wondered, and then what we did in the next study is actually changed view this dynamic heterogeneity as a factor of cell density, and then did the wound healing experiments on different cell densities and saw and also different characterized dynamic heterogeneity with this factor and saw if that affects the wound healing behavior. So I'll just very quickly show this and not take too much of your time. So we grow cells in different cell densities. So you can see when the cell density is increasing, cells are packing and becoming more jammed. This you can appreciate from the shape indices also, that when the cell density is increasing, the cells are becoming more packed, and also from the velocity, that the velocity is decreasing, as getting a kinetic arrest behavior. So we obtained the strength of dynamic heterogeneity parameter from this peak height of cells like correlation. And what we saw is that with cell density, there the strength of dynamic heterogeneity is increasing and the cooperativity is increasing. So this is not a surprise. So that we also saw that number of cells that coordinate their motion and also forces at low strength of dynamic heterogeneity is low, and when the strength of dynamic heterogeneity is increasing with the density, we have more coordination. And this coordination can also be actually appreciated in wound healing. When we did a wound healing experiment, you see that at a lower density, when the cells are not sparse, they're not very jammed, and the dynamic heterogeneity strength is lower, they are not able to coordinate. You can see that even if the movement, you can expect that even if the cells are distributed sparsely, there will be more movement, but the efficiency of wound healing is slower because the cells are not able to coordinate with their neighbors. So you can see that these orange cells are leader cells here. The leader cells are migrating and in the very good like direction where it has to go, and all the cells are migrating nicely towards the gap. But in this case, you see that the cells are very randomly moving and there's no leader follower behavior happening. This you can also appreciate from this tracking. So we did cell tracking and we saw that you see that the leader cell is migrating very nicely in the forward direction where it has to go. And the non-leaders are kind of following the leaders. But in this case where the density is very low, the leader follower dynamics is hampered and the movement is very random. So there's no efficiency of wound healing basically is decreased. This is the persistence and velocity graph where you can see that leader cell is very persistent. Also the non-leader cells are more persistent, but in the lower SDH you get persistence of leader cell and non-leader cell both is lower. So it's persistence showing that they are not migrating persistently in one direction. Also the velocity show the same trend, that there's more velocity of the cells when the cells are packed together, and the strength of dynamic heterogeneity and cooperation is higher. So let's, I'm just gonna finish very quickly, just showing you this. We also did reflection interference contrast microscopy because we wanted to see if focal adhesions are playing a role in this migration. So for this technique, I can, whoever wants to know more, I can explain, but this is a label-free technique to actually see focal adhesion in the live mode. We saw that focal adhesions are for the cells that are sparsely distributed, density is low. You see again the focal adhesion are randomly placed and you don't have the efficiency of wound healing. While when you have a density higher and the cells are packed and more coordination, you see focal adhesions orienting in for the leader cell orienting towards the wound and for the non-leader cell orienting towards the leader cell. So there is a leader follow behavior happening. This is just the angle plot that we did. Actually, yeah, that's all I have to show. Show. So this was just illustration image of this paper, showing that the efficiency of wound healing is much higher at the coordination of highest strength of dynamic heterogeneity. So this is what I'm going to do in BSSC now. I just joined like 2 months ago, and I'm very excited to start my lab here and at ISC. And I'm going to work on cancer initiation and aging, one of my interests, where because aging is a predominant factor of cancer. So when you have aged tissue, there's changed mechanics of the tissue, and that actually can impair the phenomena of epithelial defense against cancer. So I'm going to be working on how, when you age, your mechanics of epithelial cells change and how that would lead to an impaired defense against cancer and how that would lead to a cancer initiation. So this is my future interest, which I'm going to work on. If anybody is more interested, we can talk more about it, but I'm just finishing now because we don't have so much time. But sorry about the rush and thank you again for listening, and I'll be happy to take questions if anybody has.
Yeah, thanks, Madhavi, for that very nice talk. I think we have a question from Surabhi.
Thanks, Srinath. So, hi, Medhavi. That was a very interesting talk. And I just wanted to understand one thing from your talk, that have you also evaluated the effect of confinement in this case of leader cell formation that you see?
You mean, so we actually did the confinement assays a lot. Yes, we did that, but I don't have that slide right now. Sorry. I don't know what you mean by effect of confinement, but what we did was we confined the cells in this EBD chambers that we found. This very nice chamber that you can do typical wound healing assays on, and then we remove the chamber and then you see the cell migration. So we did both a scratch wound assay and confinement release assays.
So in this case, you are putting the confinement from, laterally from the Z.
Yeah, exactly. Yeah.
Okay. I was trying to understand if you have any kind of effect from if, let's say, you are confining the movement of the cells in a particular direction, or if you're confining the movement in
okay, like that. No, we haven't done that.
All right.
No. So that is an interesting thing to do, but we haven't done that. So what we did was actually we confined the cells in specific patterns because we wanted to see this leader cell regulation in the on the wound edge. So what we did is we confined the cells in patterns of like beaks and like this beaks pattern.
Right.
This is what we did. So do you want to see that? That I can show you that.
Yeah, because the curvature that you would form in the front of
yes, exactly. So we did that one.
Yeah,
that one we did.
was...
I can show you. I can show you very quickly. So this is the. Can you see this slide?
Yes.
Okay. So what we did was, because we were, our hypothesis is that leader cell formation is happening because of the dynamics in the bulk. But
Right.
there's also the studies, like you just said, that the curvature affect the leader cell formation.
Yeah.
So what we did is we confined the cells in these micro patterns where we have like biased the leader cell formation very much closer than what we would expect. So from our hypothesis, the leader cell formation should be at 200 microns because it's the force correlation length. But what we did, we biased the leader cell formation at 75 microns. And if the interfacial bias or curvature is playing an important role, then they all should form a leader. But actually what we saw instead, this is the movie, that even if you have a straight curvature or you have a biased curvature, in both cases you obtain the same length scale of leader cell formation. So that means that the collective dynamics is playing a much important role as compared to the interfacial bias, is what we saw.
I'm sorry, I may be extending my time. So did you try to like reduce this distance between the two curvatures that you created, or maybe increase the distance in that case?
We increased it. This is the minimum distance that we could create, because this is a soft lithography technique, and this is by hand we are making. Like in a clean room, I did that myself, and it was not possible to do it much closer than this for me. But I increased it to 300 microns as well. But in that case also, I saw the exactly same. So this, you can see it in this paper, in this NatureCom paper that we published. That is there. So even if you increase the distance, the final distance between the leader cell would remain the same, is what we saw.
Sure. Thanks a lot.
You're welcome.
I think in the interest of time, can we take the questions later, Shiva and
yeah, that's fine. That's fine.
Okay. Yeah.
That's okay.
Okay,
Sorry
thanks.
about that, guys.
Okay, fine. So let's go to the next speaker, that is Mithun Radhakrishna. Yeah, Mithun, you're here, right? Yes. Hi. Hi.
Yeah.
Hi. Mithun. Yeah. So Mithun, just have a brief introduction. He got his BTech from NIT Trichy and followed by an MS and PhD from Columbia. And he's currently at IIT Gandhinagar, and he's investigating soft matter systems, proteins, polymers, and other biological materials using the principles of statistical mechanics and polymer physics. So Mithun, the stage is yours.
Yeah, thank you, Srinath. So let me just share my screen.
Yeah.
So I think,
Yes.
Is it fine?
Yes, it is fine. We can see it.
Yeah. So I'll go ahead.
Yeah, please go ahead.
Okay. Yeah, thank you so much, Srinath, and all the organizers of Soft Matter India Investigators Meet. So this is, when I went to Shillong and Shimla, I really felt young. So now I'm not sure if I qualify, but anyways, thank you for still considering me young. And yeah, so today I'm going to talk about some of the work, just maybe related to, since it's a biological soft matter session, about proteins, what we have been doing at IIT Gandhinagar. So just to give you other work, what is going on. So in our group, we are also looking at, ours is mostly a theory simulation group. So we do Monte Carlo, molecular dynamics, mean field theory approaches, and to understand soft matter systems. So this is some work which is going on. We are interested in how grafted nanoparticles. So these are nanoparticles grafted with polyelectrolyte chains, how they assemble in solutions. So we are able to kind of design variety of structures, from rings to aggregates to nice sheets to crystal lattices. So this was recently published in Langmuir. So if you're interested, please go through it. So the other interest which we are also currently looking at is aggregation, and specifically looking at human gamma D crystalline. So this is the, so our eye lens contains a crystalline protein, and whose aggregation leads to formation of cataract. So we are looking at studying the unfolding pathways of it. So it's again an all atomistic simulation, and we are using NAMD, GROMACS, and some aspects of bioinformatics.
So another research that is going on is we are trying to model 3D electrodes, and we're trying to kind of engineer and model 3D electrodes for high density supercapacitor applications. So again, here we are using some principles of graph theory, and of course, molecular dynamic simulation and some free energy calculation methods. So in the interest of time, I'm not going to delve deeper on this. So just all of us know that what are proteins. Basically, these are polypeptides made up of amino acids. And we know each amino acid has a amine group and a carboxylic acid group. And how we differentiate the individual amino acid is basically this functional group R. And if my functional group is kind of an alkyl base, so as you see isoleucine, then we call them hydrophobic. And if it's basically charged, right? So aspartic acid, it's negatively charged. If it's R is CH2,4NH2, it's lysine, it's positively charged. So basically, why I'm telling you this is for the simple lattice model I use. This is all that is required. So basically, these are polymers, and they self-assemble, as we know, into alpha helices, beta sheets, and random coils. And there are many studies which show that the function is encoded in the structure, and these have unique native states. And the free energy landscape is pretty rugged. And then the concepts of cooperative folding comes in, and all of you might know about Leventhal's paradox, etc. So, and they exhibit unique thermal transitions, like the coil to globule and folding transitions, right? So these are some of the properties which I want my model to exhibit, right? So these are some proteins which I took from the data PDB from the, and what I've done is I've looked at the hydrophobic surface, right? So for the corresponding protein, these are the hydrophobic surface which I'm looking at, and the red are the hydrophobic groups, and the blue are the hydrophilic groups. So generally, if I look at a protein and if it's in solution, aqueous solution, then it's generally believed that the hydrophobes are buried inside, right? So, and the hydrophilic groups are exposed. And this is consequence of hydrophobic effect. But there are, if you look at a protein, and if I look at the surface, there are still some patches of these hydrophobic groups which are exposed, right? So, and why this becomes very important is when I look at a concept like protein aggregation. So the basic principle is I have a protein which is in its native state, and it may unfold, right? So for example, this is my protein when it is being synthesized, and it can either fold into a native state, or it can, if it comes in contact with other unfolded protein, it can form these aggregates, right? So there are some machinery, molecular machinery called chaperones. So basically what they do is they kind of encapsulate these unfolded states, right? So, and they provide a shielded environment for the protein to fold. Once the protein is folded, it spits out, right? So this is a general understood mechanism of how chaperones work. And they also play a very, very important role in sort of protein disaggregation and repair, right? So for example, if this is just a cartoon representation. So, and if my proteins are aggregated, so there is a machinery which are HSP 70, HSP 70 and 100. So these heat shock proteins machinery. So what is thought is the first step is to disentangle these aggregated proteins, right? So once my proteins are disentangled, then you provide a shielded environment for it to unfold and to fold back to a native state. And then you have a folded state, right? So we want to understand how the patches on the surface affect protein conformation or protein stability, both in case of a dilute sense when the solution is dilute, and also what happens in a concentrated regime where the proteins are aggregate prone. And there are many models, right? So currently we are mostly doing MD simulations, but we always try to understand from simple models and then try to look for that particular aspect in our MD simulation. So these are some of the common models for proteins. There are this is a 2-bead model where you have a alpha carbon, and this is the side group. Then you have the famous Honeycutt-Tirumalai model. Then you have currently you have the Martini model, which actually is showing pretty promising results when especially looking at the membrane proteins, etc. And then you have the prime model, which from Carroll Hall, right? So, and this is the HP lattice model. So this, and in the current study, we'll be just using the HP lattice model. And that is the reason why I kind of stressed on the two identities of the protein, either hydrophobic or polar, right? So in this model, it's a very, very simple model. It's a 3D lattice model. What you do is you label your protein, your amino acid as either hydrophobic or polar, right? And then you grow it in a 3D lattice. So the sequences are actually constructed using something called the core hydrophobic core algorithm. So basically what it tries to do is it tries to mimic hydrophobic effect. So for a given sequence, so not all sequences behave like proteins, right? So for a protein, what do you want to have a nice structure where you have a hydrophobic core with a polar exterior, right? And you need to also have the free energy landscape similar to that of proteins. You need to also show some thermal transitions like coil to globule and folding transitions, which are very important. And these are and you need to have a unique native state, right? So these are all the properties which are captured by this HP lattice model. So as I'm saying, I've just shown a 2D representation here. So on the left side, so you label your amino acid either hydrophobic or polar. What you say is all non-bonded interactions, if my hydrophobe are just a unit lattice away, then they interact with the favorable energy, which is -1 is just in reduced unit, right? So I'm using reduced unit, so it's some value -1, which means favorable. All other interactions are athermal. For example, polar and hydrophobic polar are athermal. So this is just if my protein is in bulk, right? And water is present implicitly, and that is the reason why you have the negative interaction between your hydrophobes, a favorable interaction between your hydrophobes. And if I'm modeling adsorption or if I am modeling a surface, hydrophobic surface, how I do it is I have a surface which has all these lattice sites, and I say that only the hydrophobic groups of the protein interact with the energy minus lambda with the surface, where minus lambda is your surface hydrophobicity. So if lambda equals 0, it just represents a athermal surface. Whereas if my lambda equals 1, it just tells me that my protein surface interactions are as strong as the intra-protein hydrophobic HH interactions, right? And we have looked at various surfaces. So if this is my fully hydrophobic surface, then I'm looking at this pattern surface, right? So these are a pattern in 2x2. The patterns are hydrophobic, and this is in 3x2. I'll tell you the reason why these are the specific patterns are chosen. These are 4x4, 8x8. There are two parameters I can change. I can either change the nature of pattern or I can change the density of pattern. For example, here this delta is the distance between the adjacent pattern. So I can change delta, which basically changes the surface density, or I can change the type of the pattern. So if I look at my folded protein here on the right hand side, you see that this is the folded state. So in the folded state, you have the 6 hydrophobic groups exposed in a 3x2 fashion, right? So there are, it's a rectangle sort of a thing, right? So it's in a 3x2 fashion, it is exposed. So in order to study sort of complementarity and pattern recognition, etc. So that's the reason why we have patterned our surfaces on using different patterns. So these are some of the simulation details, right? So you carry out in a cubical box, periodic boundary in X and Y, mimicking bulk, but in Z direction, you have a surface. So at Z equals 0, the surface is hydrophobic, and Z equals L, it is athermal, and your box is large enough. So, and we do Monte Carlo simulations at various temperatures and surface hydrophobicities. And we use a technique called weighted histogram, right? So, and from there, we kind of combine all the data from at different lambdas, surface hydrophobicities and temperatures. And finally, we are able to calculate the free energy, right? So in the interest of time, I'm not going, this is a simple, all of whoever has taken StatMech, this is pretty trivial for me. Basically, the idea is to get the free energy, and free energy, once I have the free energy of all the states, I know at equilibrium. So if the folding temperature is somewhere, what I'm saying is where there is an equal likelihood of unfolded and the folded state, right? So when I calculate the free energy of the folded state, and then I also look at the free energy of unfolded state. So this is basically here what I'm saying. So if my free energy of folded is equal to unfolded, that's my folding temperature. So of course at temperatures greater than folding, the unfolded basin is more populated, and temperatures less than Tm, the folded basin is more populated. Okay, so here there are 4 graphs, and on the y-axis I'm plotting the folding temperature, and on the x-axis I'm plotting the surface hydrophobicity. So basically, if I go from 0 to 3 on the x-axis, it means I'm making the surface more and more hydrophobic. And this data is for 4 patterns: 2 cross 2, 3 cross 2, 4 cross 4, 8 cross 8. So let us first, if I just look at 4 cross 4 and 8 cross 8, because there's not much happening there, what we see is that as I increase my surface hydrophobicity, my folding temperature goes down. And what is the reason for that? Because my protein has a hydrophobic core, even though it has a hydrophobic patch outside, but a lot of hydrophobic groups are inside the protein. So it has a hydrophobic core which wants to rupture in favor of protein surface interactions, because my surface is becoming more and more hydrophobic, right? So, and 4 cross 4, 8 cross 8, there are a lot of surface sites available. So the protein just wants to unfold, and as a result, my folding temperature goes down, irrespective of at various distances. So basically delta 2, 4, 8 just tells me that I'm changing the distance between the patches. But since the patches themselves are large enough, there's not much of a predominant effect of this distance that on the folding temperature. And it is almost similar to the behavior on a homogeneous hydrophobic surface. Whereas for small patches, where for here, if you see 2 cross 2 and 3 cross 2. So let us just look at maybe 2 cross 2, for example. What happens on a, if on a fully hydrophobic surface, which is green, I'm just written it for reference. It unfolds pretty soon, right? Even at a lambda of 0.4, it's already unfolded. Whereas if I look at various distances, right? So for the maximum stabilization is seen when delta is 8, meaning to say, if my patches are separated farther, so then what is happening is, since the protein has a hydrophobic patch that is exposed, so the protein is coming and absorbing on the surface in its folded state, and it is getting stabilized. And there is no propensity for the protein to unfold, because as it looks around, there are no other surface hydrophobic groups around. So there is no point for the protein to unfold. And as I decrease the distance between the patches for 2 cross 2, you see the transition where it becomes unstable or it unfolds, shifts to lower values of lambda, right? So for example, for my delta 8, it is around 2.8 or 2.6. Then when delta becomes 4, it shifts to around 1.7. And when delta equals 2, it shifts to almost close to 1. So what it is saying is, as I decrease the spacings, distance between the patch spacing, now the protein has a more tendency to unfold. Why? Because that when it looks around, there is a hydrophobic patch in its proximity. But still, to do that, you need to have a sufficient high value of lambda, because the protein doesn't want to break its internal HH contacts in favor of protein surface contacts, and it does it only at a higher value of lambda. And the case, actually, the situation is pretty same for 3 cross 2, except that the folding, if you look at this, the increase in the folding temperature, it is slightly higher than compared to my 2 cross 2 patch. And the reason is that a 3 cross 2 patch is exactly complementary to the exposed hydrophobic group in a protein. So as a result, the stabilization achieved is much, much higher. And this is just, we plot the adsorption isotherms, and it basically confirms what was discussed in the last slide. So basically what it's showing is, for 8 cross 8 and 4 cross 4, the proteins are generally adsorbing on the surface. So I'm plotting the protein surface contacts, right? So they are just increasing, right? Both for 8 cross 8 and 4 cross 4. Whereas for 2 cross 2, you see there is a regime where so for a fully hydrophobic, it just unfolds. Whereas there is a regime where the protein just sits in its folded state, and beyond the value of lambda, then the protein starts to unfold. And it's the same case for both 3x2 and 2x2. And for just for reference, I've just plotted here at these are at different deltas, right? Yeah. So now this is the overall picture, what is happening of the protein in bulk, in a dilute case, right? So on a hydrophobic surface, it's basically unfolding. Whereas if I have a patchy surface, but my delta is very less, even it unfolds at high values of lambda. And if I have a surface, even at low values of lambda, but my patch size is bigger, the protein will still unfold. But a case where my patch size is complementary and the spacing between the patches is large enough, then the proteins I am able to stabilize the protein. So the learnings here are that where can I apply it? So maybe it becomes very important in terms when I look at even industrially, when I look at enzyme immobilization, right? So at what density I want to immobilize, and how do I design these patches or surface functionalization, etc. But from protein aggregation purpose, also it becomes very important. So there are many other studies. This is again some of our work where we show that these are IDPs. In bulk, they actually don't have any structure, but upon adsorption on a surface, so for example, here it forms a nice beta sheet. Here it forms sort of a helix. This is, and basically surface is acting as a template to stabilize the protein. So this is just to show how this also can be. So we are currently working on studying aggregation of this cataract protein. So here, all this is a molecular dynamic simulation. So what I just want to kind of prove that point, that this is not just a model, which is sort of artifact of some model. These are commonly seen. So you have this is a structure of your human gamma D crystalline, the wild type. So it's made up of two domains. One is my NTD, the other is my CTD. And even when NTD, there are two domains, NTD1, NTD2, CTD1 and CTD2, right? So we did molecular dynamic simulations of this protein in explicit water and etc. So what we see is that on here I'm plotted the fraction native contacts as a function of time. So this is we have done up to 4 microseconds. It's a huge kind of a task when it comes to MD. Experimentally, it's nothing, but yeah. So what we see is the domains are very stable, right? Whereas individual domains, so when I do simulations on individual domains, they are very stable, as shown by there's no decrease in my native state, native contacts. Whereas individual motifs, if I'm doing it, they basically unfold, as can be seen by the decrease in Q value. So again, what is happening is that NTD1 and NTD2, they are complementary. So the hydrophobic groups are holding them together. And even in CTD1 and CTD2, it's like both the domains are actually complementary and they're acting like a surface to each other, right? So for NTD2 is acting like a complementary surface for NTD1 and vice versa. So so although it's not the aim of this project, we are trying to study aggregation. So, but this is a common, it's not an artifact of the model. So using the same thing, we wanted to now, of course, study aggregation. So aggregation, of course, happens in higher concentration. So to model that, what we did is we took, instead of one protein, now we take two proteins and we look at them at two regimes. So the first regime is called the isolated regime, where the hydrophobic groups of the two proteins don't interact with each other. And then we look at something which is a concentrated regime, where the hydrophobic groups of the two proteins, so there is a inter-protein interactions, they interact with the energy -1, which is almost equivalent to the intra-protein hydrophobic contacts. So what we see here is, if you look at the left-hand side, I have plotted the total energy, right, as a function of temperature. And what we see is the total energy of my non-isolated proteins, where the proteins are able to interact with each other, that is always less compared to that of my isolated proteins, meaning to say the free energy, when I calculate the free energy, is always lower than that. Whereas if I just look at the protein itself, neglecting the interprotein contacts, just the protein energy, which kind of signifies how folded my protein is, my isolated proteins always has a lower energy, showing that it is more folded, it is compared to my non-isolated proteins. So non-isolated proteins basically misfold, and because of the inter-protein contacts, and they are actually they cannot fold back. And there is also many theories that the proteins even inside our body, they may be just marginally stable, right? So even a small change in any of the external environment leads to protein unfolding. So now we wanted to kind of look at, can I use surface again to first, so because the first aspect of protein unfolding is to disaggregate them. So what we did was, okay, I took a hydrophobic surface and we measure the interprotein contacts at different temperatures. So what you see is here, as I increased my temperature, the interprotein contacts go down. So why do they go decrease? Because now there is a competition between protein-protein interactions and protein surface interactions. So if my protein surface interaction, if I'm able to kind of increase it, if I can increase it, so then you see sort of a transition sort of a thing occurring, and I'm able to sort of mitigate the interprotein contacts. But this is still not complete. As you see, for our model protein, there is still sort of 5 contacts which is still there. That's because the proteins, even upon adsorption, also can interact. So they can adsorb close to each other and they can interact. So now to see what happens, what we did was, yes?
Yeah, can you quickly wrap up?
Yeah, I'll wrap up in 2 minutes. So now to understand the effect of patchy surfaces, what we did was, now we templated our surface into patches. So this is, for example, this is at a distance 8, meaning to say the patches are farther separated. And there are 3 graphs, right? I'll just concentrate on this slide. So the first graph is the number of hydrophobic intraprotein contacts. So this is the first graph. The second graph is the protein surface contacts. And the third graph is my protein intraprotein contacts. So my objective is what? There's a twofold objective. First is to decrease the interprotein contacts, that is to mitigate protein aggregation, and then to kind of enhance the protein intraprotein contact, making the protein more stable, right? So as you see here, these are for different patterns, and each of the pattern, so on a fully hydrophobic surface, there are some contacts still there. Whereas if I make the surface patchy, I am able to completely mitigate intraprotein contacts, because these proteins are coming and maybe absorbing on different patches. So I'm able to completely mitigate them. But whereas again, and the result why they are mitigated, as you see, is because of there is an increase in protein surface interactions. So proteins are adsorbed. That's the reason some of the intraprotein contacts are mitigated. But what is very interesting here, okay, the dotted line is the behavior of a dilute protein in bulk. So remember that. So this is the behavior of dilute protein in bulk. Now I'm calculating what are the hydrophobic intraprotein contacts as a function of surface hydrophobicity. So what you see is, for a hydrophobic surface, it just decreases, meaning to say the protein just unfolds. So even though our first objective is achieved, but the protein is getting destabilized. Now, whereas for patchy surfaces, we do see an increase in the intraprotein contact. That means that patch is kind of acting as an anchor and helping the protein to kind of fold back to its native state. But what is very interesting is, in case of blue and red, which is for 2x2 and 3x2, I am even able to stabilize the protein more than what it is in my dilute solution, right? In the dilute case in bulk at that value of lambda, right? At lambda equals 0. So not only I'm able to disaggregate proteins, I'm also being able to stabilize the protein. So this was published in PCCP as a hot PCCP article. So basically just shows that at 4 and 2, we don't observe that behavior. So this is overall what is happening, right? On a hydrophobic surface, they are adsorbed, but still there are interprotein contacts. Whereas on a patchy surface, the 2 proteins are adsorbed in separate patches, and I am able to achieve both disaggregation and stabilize the protein. So just with that, the conclusions are pretty trivial, what we have already done. So we have also developed this tool. I kindly encourage all students and also research staff and faculty to explore this. So basically the idea is if I enter a PDB and it gives lot of all the properties of the proteins. In the interest of time, I'm not able to show it, but please explore. And this is a module in development, so we are trying to build more. So with that, I want to thank the funding agencies and thank you for your attention. Yeah, thank you.
Okay, Mithun, thanks for that wonderful talk. And we have maybe time for one quick question. Shortly, we are running short of time today. Just, one quick question. Yeah. Okay, I had a question about this one. So you are changing the size of the patch and the distance between patch. So just so you can fundamentally design that substrate to achieve any kind of property that you want for your chaperone, right? Is that
yeah. So the idea is that because with enzyme immobilization, for example, if I look at that, so then you need to have throughput for which you need to have enzymes immobilize at a particular density. So you cannot compromise. If I just have two enzymes at kind of situated farther apart, then even though it might work well, but your throughput will be less. So there is a kind of optimum value where you want to kind of stabilize this, right? Functionalize them. And with chaperones, if you look at the structure of chaperones and the presence of, so there are these hydrophobic patches which are present in chaperones. It's experimentally a lot of papers which have come recently, and this the simulation very nicely kind of supports the mechanism, right? So what is happening, etc.
Okay. Thanks, Mithun. So we will go to the next talk.
Yeah, thank you.
Okay. Was I not audible? Hello? Can you hear me?
Yeah, we can hear you now.
Okay, sorry. Yeah. So a warm welcome to Dr. Surabhi Sonam, who's currently at the D.Y. Patel University, and she did her BTech in bioengineering from Shastra University, followed by a PhD at Mechanobiology Institute at NUS, and she did her postdoc at Institute of Jacques Monod in France, and she's today here to give us about her new work that she has been doing. Thank you, Surabhi.
Thanks, Srinath. So thanks for the kind introduction. I'll just share my screen quickly. All right, I hope you can see that.
Yeah, it's coming up.
Yeah.
Yeah, we can see it.
Sorry. Yeah, all right. So what I'm going to, I'm sorry, my computer is hanging for some reason. I'm trying to figure it out. Okay, I'll see. 1 second. Yeah, all right. I hope you can see it.
It's not in full screen mode though. At least I don't see it.
It just crashes as, I mean, my computer is hanging up as soon as I go in full screen mode. Can you just give me 1 second? I'll reconfigure this. Yeah.
Yeah. Okay.
Sure. Sorry. Okay. All right. Yeah. Okay. Is that okay now?
we can't see it yet. Yeah, now it's.
Yeah, so unfortunately, as soon as I go on full screen, it just crashes. Can I join with another machine?
Or maybe you can just start with this. I mean, can you change your slide and let's see what if the size is good enough, then maybe we can start like that only.
Yeah, my screen is just going blank. So I'll just use my tablet very quickly, if that is okay.
Okay.
Yeah, sure. Thanks. I'm sorry, I don't use Zoom very often. We are normally used to Google Meet. And I don't know if that is the problem.
Not a problem. Alternatively, you can also send any one of us your slides and we can also share it in case you feel that might be.
Yeah, that could also work. If that is okay, I'll send it to Sunita, if that is okay.
Yeah, that should be fine.
Sorry, since I have only your email address.
Okay, we can see it now.
Yeah, we are able to see the slides. Yes.
But we can't hear you.
Muted, sorry.
Yeah, I'm sorry. I as soon as I go on full screen mode, it just.
Okay, maybe just send it to Sunita and then we can.
Yeah. I'm uploading it on Drive to share. Sorry for the delay. Maybe I'll try to shorten my talk so that we do not exceed the time here.
Of all days, our laptops and iPads work perfectly fine.
Yeah, I know.
And when we want them to work perfectly, then only we have all the glitches.
Yes, I agree.
Yeah, if this takes actually time, like I think what possibly can do is like you can just put this PPT in one of the organizers thing in the chat box. They can present and then you can just speak.
Yeah, Surinder, that's what we're trying to do.
Ah, okay. So sorry,
No, that's okay.
Okay.
So I'm giving a one final try from my computer, which is not working. Sorry, my computer has decided not to work finally. So I will quickly hoping that it will get uploaded on at least one of the platforms, either Dropbox or Okay, fine.
There
So
are no animations. Maybe you can convert it to PDF and then it will not have any problem present.
yeah, unfortunately, I have a lot of videos.
Okay.
So I will join from my tablet now.
Yeah, that's why maybe uploading is also taking time.
Yes, I know. Yeah, sorry. Sorry, I hope I'm audible from here now.
Yes.
Yes, you are.
Okay, so let me share my screen. Sorry, for some reason my tablet is also not helping me. So I have managed to send it to Sunita now, and hopefully Sunita has received my email.
Let me check.
Yeah. Meanwhile, I'll try again to share from my computer if I can.
It says you have to give me the access, Surabhi. It says access denied.
I gave you the editing rights, if I remember, if I understand well. Can you request for an access?
Yeah, I did that.
Already. So yeah, you're an editor now.
Yes, I can see it here now.
So anyway, meanwhile, Sunita is able to share the screen. I'll just try to give an introduction of this entire field. What we might hopefully will be able to see is
it will be like in another maybe 10-20 seconds visible.
Yeah, sure. So I'll for the essence of time, I will try to give an introduction in the meantime. So I'm really sorry for this delay of 15 minutes, and I'll try to finish it off in another maybe 15 minutes as well for the essence of time. And what I'm going to talk about is essentially as an effect of forces, the different kind of behavior that we might see in case of an epithelial monolayer. And sorry, so yeah, thanks Sunita. So all I'm presenting here is the effect of mechanical stress driven by rigidity that governs epithelial stability. This work has been done in collaboration with Benoit Ladoux and René-Marc Meche, who are at Institut Jacques Monod at France. So this is my postdoctoral work that I'm going to present, and it is still unpublished, so we are under the process of review, and that's why most of this work may be not familiar to anybody right now. Anyway, so maybe we can go to the next slide here. And this work has, I mean, there are multiple people involved in this work, including people from Singapore, from MBI and US, and then from other parts of the France and CNRS. And can we go to the next slide? Sorry, yeah, thanks. So at cellular scale, generally, we see a lot of forces acting inside the epithelial monolayer that can create many different kind of effects, as you can see here. So they can all generate different kind of flow inside the epithelial monolayer as well. You could have different persistent lengths of the movement of the cells. Again, you could have leader cell formation, as was highlighted by Medhavi just like couple of talks ago. So what these forces tend to do, can we go to the next slide, Sunita? Sorry. Yeah, so what these forces generally tend to do is to create different kind of compressional behavior in the tissue. Let's say what you see on the left side here is compression behavior inside the tissue, and that can lead to eventually phenomena like detachment of the cells from the monolayer, eventually leading to extrusion. And on the right side, what you see is development of a local tension, which could lead to division of cells, as has been shown in the bottom image here from in this paper. Sorry, can we move to the next slide? Thanks. So using these kind of behavior, cells tend to move in different directions, and also that leads to generation of tension within the monolayer, which changes along with the point of movement to the bulk of the cells. So if you see this paper from Trepa, which was published in 2009, quite a landmark paper for this field, is it shows that there is a change of stress within the monolayer from the cell, from the tissue periphery to the center of the tissue. And if these kind of tensions increase further, it could lead to the rupture of entire tissue. Sunita, can you press again? Sorry, thanks. So in this case, you can see that there could be a rupture of the entire tissue, leading to really large hole formation. And this is how actually one of these organisms migrate, that they create holes inside themselves, then they heal it, and then they create. So it's a spontaneous hole formation that you would see. So we try to understand this kind of behavior. Thanks. Can you go to the next slide? Thanks. So we try to understand this behavior, and just to simplify the entire system. Of course, it is a much more complex system that we were looking at. So we try to simplify it by creating these deep UV patterned cover slips coated with fibronectin, transferred it on gels of different stiffnesses, and then we added MBCK cells on top of it. So what we realized, can you just press the next button? Thanks. So what we realized, based on the stiffness of the substrate, you could see these hole formation in different kind of substrates, and it is limited by the stiffness of the substrate. So as you can see on the left movie at the bottom, there are hole formation, very clear hole formation, spontaneous also, and then they some of them heal themselves, some of them don't heal themselves. However, if you see at the second movie on the right hand side, which is 55 kilopascal, which is a 55 kilopascal movie. So in that case, you can see that the cells do not tend to form any kind of a hole. So in that case, basically, what we think is the stiffness that is driving these hole formation. And when we went on to look at the role of the stiffnesses, actually in this case, what we see is that as we increase the stiffness of the polyacrylamide gel, the number of holes decrease in each of these circles that we are looking at. And of course, it is not limited to only MDCK cells. If you look at the right movie on the right hand side, which is a movie of a CACO2 cell, and in that case, you can see that again there are hole formation at 2.3 kilopascals. So it is not limited to only MDCK cells. Can we go to the next slide, Sunita? Thanks. So the first question that you might ask is that it's possible that the entire surface itself might be getting degraded, or let's say on the surface, when you are whatever fibronectin or ECM protein that you are adding, that might be getting detached. But so when we sorry, the movie on the left, sorry, the first movie. Okay, thanks. So if you look at this, you can still see that the fibronectin concentration stays almost the same on the surface. However, we still see hole formation in these movies, and these holes, as I mentioned earlier, could be of two different kinds. One type we see is a short-lived holes. They tend to form, they tend to close themselves. However, some of these holes can tend to live really long. So when we say really long, which we are considering more than 5 hours in time scale here, and then we try to understand why these holes might be forming. Can we go to the next slide? Sorry. So one first thing that we thought of is possibility is adhesion. Of course, it's not just the availability of fibronectin molecules over there, but also the possibility of adhesion here. So what we did is a very simple experiment to understand the bidifferential coating of fibronectin. So in the center, if you look at it, we used a system called Alveol. Sorry, it's a company's name, Alveol. They have a machine called Primo, and we use this machine to have a differential coating. So in the center of the bottom left image that you see, we have fewer molecules of fibronectin. So it's the lesser density of fibronectin, and at the sides we have more concentration of fibronectin. So when we put cells on them, if you could play the movie on the right side, thanks. So if we seed cells on them, they tend to form holes at the center where the fibronectin concentration is lesser than the sides where the fibronectin concentration is really high. Can we go to the next slide? Sorry. So in this case, the first question, the second question that people ask is that could there be a role of the density? As we could see earlier, the holes that are formed at for long-term scale, they are formed at a higher density. So what we did was to abrogate the division of the cells using mitomycin. And can you just play this movie? You can still see hole formation. However, the number of the holes that are forming on these circles reduce. So that can be seen by the graph on the right side. So this again, density has a role to play here with respect to what we were seeing earlier. And in general, what we saw is that whenever there is a hole formation at the edges, we have these very, very stretched cells. So that you can see with the zoomed images on the right hand side top, and you can see that they are extremely stretched cells, and that can be also that we quantified with the angle of the cell with respect to the curvature of the hole. And if we remove, so in the simulations, what we did is to remove the active stress, and if we remove the active stress, then the number of long-lived holes reduced. So this is an active stress which is pushing the boundaries of the holes and keeping it intact over there instead of allowing them to close. So experimentally, sorry, yeah, we can go to the next slide. So experimentally, what we did is to use blebistatin to reduce the tension within this system, and what we see is that the hole formation is very small. There are fewer holes. The sizes of the holes also do not increase as much. and then it would be very difficult if you do not get used to this movie. It would be very difficult for you to identify where the hole is forming, because the sizes now go down to one cell. To the scale of 1 cell size or to maximum 2 cell size. So this definitely is coming from the tension within the tissue or the tension around the tissue. Can we go to the next slide? So what we did from here onwards to look at the tension within this tissue, and since for the essence of time, I will not explain what TFM is. Thankfully, Medhavi has already explained what TFM is, so I'll skip that part, and I'll go to understand what is happening here. Sorry, yeah. So what we did is to understand if there is stress formation inside the tissue or not, and if there is an intracellular stress here or not. And what we see is that in the confined and both unconfined monolayers, we have an increase in tension within the tissue. What you see on the top is a traction force. So the traction force is reduced inside the confined molar layers. In both confined and unconfined monolayers, and for the soft gels. Sorry, and if we take the isotropic stress, which you see at the bottom, you see that there is an increase in the tension within the monolayer, and there is a compression-like behavior when we go to the stiffer regime of the substrate. So at 30 kilopascal, we see that there is a compression within the tissue. When we did the same experiment with laser ablation, what we see is that the recoil velocity of the entire tissue reduces as soon as we go to a higher stiffness. So this clearly shows that there is a tension within this tissue at a softer substrate with respect to what you would see in case of a stiffer substrate. And using this information, what we did eventually to look at when this tension is developing inside this tissue, and actually if that could be the reason to actually lead to the hole formation. What we see is that roughly around 35 minutes before the hole formation, there is a development of tension inside the tissue. So you can see on the left side there is an increase in tension at roughly 35 minutes before the hole formation, which reduces as soon as the hole opens up. And it's the same thing that has been plotted in the graphs, that you can see that there is an increase in the tension before the hole formation. Can we go to the next slide? So since there is a combination of both compression and tension inside in tissue, normally what we see, this kind of behavior normally leads to interesting kind of defect formation that you normally see in case of crystal substrates. So if you compare the tissue, consider tissue which is as polarized objects, something that is called nematic objects. So if you consider them like nematic objects, you can see different kind of defect formations here, and these defect formations have been plotted here on the top and the bottom, and the left side of the top and the bottom, and you can see that there is a plus half defect. So that is one kind of organization that you see, and minus half defect, which is another kind of organization that you may see, and that has both of them have a different kind of organization of both compressive and tensile regimes inside them. But when we look at the hole formation with respect to this compressive and tensile regimes that are formed by either of the kinds of defects, what we see at the bottom of the right side bottom, what we see is that most of these holes are formed at the extensile regime. So maybe you will be able to appreciate. Sorry, can we go back? Yeah. So maybe you'll be able to appreciate from the images that there is a higher tension at the location where the hole is forming, which has been marked with blue, and also in the bottom image it has been marked with green. So you can see that that's where there is an increase in tension, and if we go further, take it further, what we see is that most of these holes are forming at the minus half defect. That's where you see more extensile behavior. So at the center of the minus half defect, which is where we see most of these extensile behavior. So can we go to the next slide? I would just like to conclude here very quickly now that what we see is that there is an increase in tension in the tissue because of the topological effects, because of the topological organization here, as I was discussing earlier, and that could lead to the hole formation, which eventually is elongated because of the extensile stress, which is present in the tissue because due to the surface and the cell interaction, and that could lead to two different kind of hole formation. One is a long-lived hole, another one is the short-lived hole. Some of these long-lived holes and the short-lived holes can eventually close, and in some cases they tend to form only fingers, and then they stay just like that. So this is where I would like to conclude, and I would thank all my collaborators and the lab of cell and adhesion mechanics and all the funding agencies here, and thank you very much for your patience and your time.
Okay. Thanks, Surabhi, for quickly wrapping it up, and then you didn't exceed your time at all, even with all the problems that you had. Okay, so I think we have time for a quick couple of questions. I think Shiv Sundar would be.
Okay, thanks. So it's excellent work and excellent talk, Shruvi. So I really enjoyed it. So I was just trying to understand the differences between the stiffer substrate and the softer substrate, right? So in your when the elastic modulus of substrate is lower, so then your holes are forming. So how do one understand this? So is there a role that the friction plays here?
So theoretically, if we say so, yes, that's what we expect, that there could be a role of friction. There could also be a role of because of the porosity of the gel, because we are looking at gels here. So the gels have different kind of porosity if we go into softer regime, and in that case there is also change because of the gel porosity. There's change in the density of the adhesion molecules that are present at the surface. So that could also lead to this kind of hole formation from the substrate side, since we are looking mostly from the substrate side here. So but then that eventually also is coming from the cellular side, that if we look at the softer gels, I'm not sure if you're familiar with this area of research, but then normally there is change in adhesion. So the focal adhesions tend to become smaller on the softer substrates, and that leads to very weaker adhesions. So as we go, as the density increases, something that I have not shown here, but as the density increases, what we see is that the adhesion focal adhesion becomes even smaller than what you would expect in case of softer regime, and that becomes slightly easier to be broken with respect to what you would see in stiffer substrates, because the focal adhesion is still slightly bigger than the softer than what you see in softer regime. So it's I would say it's an interplay of both kind of things that we are that might be leading to the hole formation.
Okay, thanks. I still have questions, but I think I would catch you up maybe later at some point.
Sure.
Thanks.
Yeah.
Okay, thanks. The last question for the speaker, Vibhu.
Yeah.
Go ahead.
Hi, sir. So excellent talk. So yeah, I have actually two questions, but I will say how it goes. What Shiva asked about this soft and stiff substrate. So I also assume that it's kind of a fracture you are doing, and you need to exceed some sort of a threshold contractility of the network, and that can happen only beyond certain stiffness. I mean, if you look at the individual cells, I mean the contractile network kind of are more formed or more prominent if you have a stiffer substrate. So I mean, can you relate this from an individual cell to a monolayer? I mean, are you also thinking of in that line, because your stress fibers are more prominent on a stiffer substrate, and then on a softer substrate they are not able to hold that contractility as much as you expect, and you need some certain threshold of contractility to form the holes. Is it am I going in the correct line?
So I understand what you want. I mean, if I understand well, what you mean to say is that since there are stronger stress fibers, you would expect, I mean, in the stiffer regime, you would expect them to apply more forces within the cellular monolayer, and that should lead to the fracture or hole formation, as we are calling it here. This is slightly different, I would say, because in single cell, what you see is there is a more likely formation of this beautiful stress fibers. And in single cells, again, if we look at single cells which are on softer regimes, they do not spread as much. So that is a different kind of a behavior that you would see. But as we go into a bulk, the behavior of cells changes slightly, because in that case, again, in the bulk, you see that the cells do not form, even on stiffer substrates, cells do not form as nice or as long stress fibers as you would expect for a single cell, or at the periphery of this entire monolayer, if you have a periphery, let's say. So that would have a different kind of a behavior with respect. I mean, the single cell behavior that we see is very different in terms of the tissue behavior when we look at soft and stiff substrates. But as I mentioned earlier to Shiva also, that it comes from both directions, that there is a weaker adhesion thanks to the I would say crowding of the cells, and also because the less forces are being applied between the substrate and the cells at the softer regime. So that also makes the entire feedback mechanism weaker, and eventually you have weaker interaction between the cells and the substrates. So that becomes slightly easier to break. What we have seen very, I mean, I would not say it's very conclusive, because these holes form very, very quickly, and unfortunately, even on confocal, we could not catch it very well. So what we saw very roughly is that most of these hole formation begin at the bottom of the cell adhesion, and then they propagate further. So that's why I said that there is a breakage of the cell substrate adhesion before the cell adhesion. And when you have more when you have more stress fibers to actually lead to a higher increased intercellular stress, that should break the cell adhesion first, and then maybe eventually break the bonds at the cell substrate adhesion. Yeah.
Right. Yeah. Thanks. I have some more questions, but maybe
Yeah,
we
can discuss. Yeah, sure.
Okay. Fine. Thank you, Surabhi, and all the participants for asking very good questions. So I think we are done with this session here.
Yes, so I think so now we can open the floor for maybe 10 minutes of breakout rooms, Danny.
Yes, how many minutes do you want? I can time it.
So the mini talk should start at 11:45. So we have around 10 minutes maximum.
Okay, so I'll.
But the first speaker is not there, right? So I think we should have some extra time.
Oh, in the mini talk?
Yeah, it's not. I mean, when I see the list of the mini talks, it's like missing.
No, his title is missing.
Title is missing, Surinder.
He's there.
Okay.
No, we had one dropout yesterday's session, but today I think we have.
Okay, excellent.
So Shobra, 10 or 15?
10 minutes is fine, so that we stick on time.
Excellent. Okay, I'm doing it right now. Yeah. Rooms are ready. You can.
Okay, so like yesterday, all the speakers can take their own rooms, and then people who want to still continue having discussion can enter these rooms and do that for another 10 minutes for the next. Danny, you're here, right?
Yes, I'm here.
So in 11:45, you will then start the along with Dr. Surendra, you will start the chairing, right?
No, the new list is different, right?
Where is it?
Shobana had sent an email for who's organizing.
Sunita, sorry.
Yeah, Sunita, sorry. Uh, let me find that.
Day to schedule mini talks. Okay, that is only for
So this is under the title, the request to evaluate mini talk session. So it's going to be Srinath, Surabhi, Siva Surender, and yourself.
that is for voting. I mean, that is for the what is it for scoring the rubrics. But to chair the session, your name along with Surendra is there. So that is what.
Okay, so I thought this was the list of both the chairs and.
Yeah, because this was what was followed yesterday. Can you check with Sunita if she's maybe spoken to Surendra or Srinath about it? Maybe to Surendra itself. Because the name just before our name, the organizer name is usually the chair.
Yeah.
At least what happened yesterday. That was what happened yesterday.
So judges for mini talk is Sheena, Surbi, Surendra, and myself. And for chairing the mini talk session, Danny's and Surendra's name is there.
So Priya, who's doing the chairing for the coming session?
You're asking me, Danny?
Yeah, so who's doing the chairing for the coming session?
That is what your name and Surendra's name is there for chairing the mini talks.
That's right. But Priya was just saying, right, that it's changed because I am chairing the session tomorrow. But I can do it. It's not a big.
So I think so. The mini talks. Okay. The schedule that Sunita sent today. That for the mini talk session, your name along with Surendra is there for doing the chairing of mini talks. That is, I think so, irrespective of whether you will be sharing a proper session tomorrow, like for invited speakers, I guess. Let me get hold of Sunita once, okay?
Sure. Can we all meet in that organizers lounge? That's another breakout room.
Yeah.
Okay.
I will go there.
Siva, you'll take care of chairing the session, right? Danny here.
Sure, that's fine. So let me just open up the list of speakers.
First one is titleless.
Hello.
Yes. So.
Hi everyone. Hi everyone.
Okay, I think thanks. So we'll start with the mini talks. I think right away it's already time. So the first speaker is Ramu Yadav, and we don't have his title. So hopefully he has his title, and we'll listen to it.
Okay.
Yes, go ahead. You can.
Hello everyone. Can you hear me?
Yes.
Yeah, sir. So I am Ramu Kumar Yadav, a PhD student at IISER Mohali. Today I will talking about the unzipping of a double-stranded black copolymer DNA by a periodic force. So the motivation behind my work is that the unzipping of a DNA is unzipping of a DNA by force is the initial step in the biological process like the DNA replication and the RNA transcription. So now first I will model my DNA. So we model it as a black copolymer. First I will model two hetero black copolymer. So we model a black like from the two different types of monomers A and B types. So we take the A number of monomers of A type, B number of monomers A type. The total number of monomers in one black is the 2n. So here we have the black A and Bn. So when we repeat this black M times, so we get a complete heteropolymer chain. If the n is the total length of the chain, it means there is n monomers in the chain. Then the number of blocks in that chain will be n upon 2n. So now we use that. Now we use two different chains to form our DNA on a square lattice. D is equal to n plus 1. N plus 1. We form our the DNA here. So while farming, we take care of the considering the nature. These polymer they cannot cross each other. Neither they cross the mutually. From the directional nature of talk, we'll decide their self-evidence and the self-evidence. So now what I do, I model my DNA here from the A and B in times. The my DNA here. So we keep the one end of the DNA fixed here. So fixed here. So when a type of monomer from one chain is in contact with the a type of monomer from other chain, so our system gains the energy of -2 epsilon. And for the B types of the base, this gains of energy in the 3 epsilons. So here you can see that this is the my DNA. So now we have the A and B and DNA, and of the length n and m number of blocks in that length then DNA. Now what I do, we keep one end of the DNA fixed here, and we keep one end of the DNA fixed here, and we apply the forces on the end monomers, and the forces, we take the absolute value of force. It is sinusoidal force G is equal to G. You can see from here, this is the sinusoidal force G naught sin omega 2. So this is because if they will not cross each other. So now for the fixed DNA, A and B N type, you can see that if I fix A here and apply the force at the B type of DNA, then there is also a DNA sequence of DNA type B and A. In one case, B will be fixed here and force will be applied on the A type of the base, and another its opposite will be the A will be fixed here and force will be applied on this type of base. So now we have two nature of forces. Now we can apply the static force here and one in the dynamic force here. So now, and we monitor the distance between these monomers. The distance between these monomers is the response in our case, and we monitor this distance as a function of the force. So now I have the we have the recursion relation of the partition functions. So from there we can use the exact transfer matrix techniques to calculate the separation between the two end monomers. So we calculate that separation as a function of the force. So we here are the three different DNA. You can see that here
You have about a minute to wrap up.
Huh?
You have about a minute to wrap up your talk.
We have here the three that force versus extension plot and a nice data collapse is observed for a critical value GC. We do it at 4 different temperatures, and then we plot this GC from an analytically observed result using the generating function techniques. So we see that GC observed from this equation is exactly matched with the GC observed from these equations. So now come to the dynamical part. The dynamical parts. In dynamical parts, we monitor the extension xt as a function of GC. So in periodic case, this shows a fluctuation. But when we take the average over 10,000 cycles, then we plot that 10,000 average x and g. Then we get a hysteresis loop. Hysteresis loops because there is a time lag between our response and the applied force. So now we have the different sequence of the DNA. So we just want to see that what is the effect of the black nature of the DNA on the dynamic case. So we see that for the chance of the smaller black length, the results were not different between the AN, BN and BN type of the sequences. But when we increase the length of the black length, for the chain, suppose we have the chain length 256. So for 128, there will be only one black. So for higher for the higher black length, we can see that there is a opposite sequences. So the difference hysteresis loop. This is in the high frequency range, and this they also show the very much difference in the lower frequency range.
Ramu, I think you should close the talk.
Just 30 seconds. 30 seconds. Okay. So now we use the numerical techniques to calculate the area of these loops. So now area of the loops. So now we plot the area of the loops as a function on log scale as a function of frequency. So we can see that the opposite sequences for the smaller block size does not show any difference, whereas the for 128 B12010 higher block length sequences, we can clearly see a big difference between the area of the both the sequences. And the intermediate sequences show the mixed results of the lower block size and the higher block size. Okay, thank you. This is the conclusion. In conclusion, I can say that in the static case, the results do not depend on the sequence, where in the dynamic case, our results depends on the which block sequence we are using. Okay, thank you.
Okay, thank you. And the discussion will be later after the next talk. So the next speaker is Samapika Sahu. Do we have Samapika here? It looks she is not here. Samapika Sahu.
I don't see her name in the participant.
Yeah, exactly. I don't see in the list. So I think then what we can do is we can continue with the discussions for Ramu's talk. So anyone has questions for Ramu? Yes, Tripta, please open your mic. And
Ramu, can you just summarize your results because I think I missed it. So what is the main?
So, ma'am, as I told, I am using different number of sequences like A4, B4, A8, B8, and A16, B16 for a fixed length of DNA. So, what I have shown that in static force case, all the sequences show the same result when we apply the static force on the DNA. But in dynamic case, when our force is the dynamic, G is equal to G naught sin omega t, in that case, our results depends on the amplitude of the force, frequency of the force, as well as on the type of the sequence. So this is the basic summarization of my work.
Yeah, but this is something you are saying mathematically, but physically, what does that mean? Like if it depends on the amplitude, do you have a, this is a simulation that you did, right?
Yeah.
So I understand what you're saying, but physically, what does that mean? If it depends, so let's say I have a DNA and you're pulling it, and then you're applying some force, and depending on is it static or if it's dynamic, you have different responses, right?
Yeah, yeah.
So
Yeah.
yes.
I exactly don't know the exact dynamics, but our system follow the Rauch dynamics. So the results is different because in one case I am unzipping the DNA from the 3 epsilon case, and one case I am unzipping the DNA from the 2 epsilon sides. So if there is a block, suppose if I am unzipping the DNA from the 3 epsilon, there will be 128 3 epsilon bases. So once they are broken, then the alternative sequence will be 2 epsilon, so then will be easily broken. But in case of 2 epsilon, when I am unzipping from 2 epsilon, so in that case, if we compare, the number of broken bases will be different in both cases if all parameters are the same.
Okay, thank you so much.
Okay.
Okay. Any other questions? If none, so I have a question. Ramu, could you please explain to me, like, just I have a naive question. So what's the reason reason for the time lag between the applied force and the response of your polymers?
Sir, we always know that whenever there is when you go to the microscopic detail of the system, you will find there are the meta-stable states. The microscopic reason behind the formation of the dash line is the meta-stable states.
What are these meta-stable states?
If suppose your system stuck there for this is very complicated. Now I am just doing the, when I will write the Landau potential, whatever, then we can say that what is there. But always the microscopic reason behind the formation of a hysteresis are the meta-stable states. So suppose your system is stuck there, it will take some time to respond, then already there is a lag. So
that's why I'm asking you, what is the reason for this time lag?
Sir, yeah, that is a meta-stable state, sir. That meta-stable state, your system will be there, and when we give in efficient force and frequency, then it can cross that potential wherever is there. Then there will be a lag.
Okay. Okay. So is there any other questions? We have a minute. Okay. So if there are no questions, let's thank Ramu. And we did not have the next speaker, so we can go now to Vignesh. Mr. Vignesh is here.
Oh, yes, sir. Hi.
Excellent. Hi. So, so Vignesh will speak about investigation of tumbling and flagellar polymorphism of E. coli using simulations. Vignesh, please go ahead. You have 5 minutes to speak, and you may start.
Okay, thank you, sir. Can you see my slides? Hello?
Yes, yes, yes.
Okay. So the topic is, as I told, investigation of tumbling and flagellar polymorphism in E. coli. Yeah. So we all know that, E. coli are a pretty interesting species to study, especially the locomotion, has a lot of relevance in biology and biotechnology. Obviously, E. coli, we know that they can cause infections. They have pathogenicity, as well as E. coli are pretty simple to study in terms of their physics, in terms of their biochemistry and their genetics. It's pretty simple to study, reportedly from biologists and biochemists and all those people. My particular work actually involves a lot of simulations. One reason for that is that experimental study is pretty challenging of these species because of the size, obviously. Like the cell body is like 2.5 microns by 0.8 microns in dimension, spherocylinder, and its flagella are like thickness is like 10 microns by 100. That thin. So it's difficult to capture in the video. these things, as well as typically, right? Typically, the microscope focuses on a 2D plane, and exactly when it's putting the 2D only, we can capture the whole thing. If it's like moving out of plane, then that's lost. You can see like in these two GIFs, like the left side, it's pretty clear. The tumbling motion, classically speaking, the one flagellum comes outside and it goes back in. Right side, no flagellum comes outside, but still there's like direction change happening. So right side is like pretty subtle, something is happening which we can't really see experimentally. So that's where in both cases, but like especially the right side where simulations come in really useful. So going to the next slide. So our model, my PI Tapan, Dr. Tapan explained yesterday, I believe. Here's another just overview of that. So we have a discretized flagellum, as we can see here. So this is ferrocylindrical cell body. So we take into account, for every discrete element, this is the equation. We take into account elastic forces, steric interaction, thermal noise, as well as the hydrodynamic interaction. This is like linear, obviously linear equation is the rotational equation. In our model, actually, we neglect the thermal noise for the flagella, since that'll make the computation much expensive. Below you can see like what is called the extended Kirchhoff free energy density. So we have taken harmonic potential based off of Kirchhoff's rod theory. And since we have 4 polymorphic forms, only 3 of them are experimentally observed, but then the 4th one is predicted by molecular molecular theory and classical mechanics theory. So these are like the 4 wells of them. So like it could be, at any stage, the flagellum could belong to one of these 4 wells, according to which its ground state configuration would be different. So I will move on to the next slide now. Okay, so our work is like, to start with, it's a pretty complex system. So getting very exact sort of patterns is pretty challenging. We are starting off with getting the patterns of like tumbling statistics. We're seeing if we can explain why the particular order that's observed, like normal to semicoil to curly, which is the experimentally observed order, what was experimentally clearly observed, why that is being observed. We're trying to explain it, perhaps seeing if that is the one that can if we computationally simulate it, if that is the one that can produce the steepest sort of direction change when the tumbling happens. So that is what this is about. Go to the next slide. You can see like these normal semicoil and curly, these are actually the same contour length, but then you can see because of the different pitch and the different radius, it's like looks different, looks shorter or longer or whatever. Yeah. So next, next is So
you have about a minute to wrap up, Vignesh.
Okay, thank you. So we see that right. So we've done studies further, not just 2 flagellum, but also with 4 flagella, and just put the video here, and we see that depending on which flagella we rotate one at a time, we reverse rotate the flagella. Depending on which we reverse rotate, it takes different times for the tumbling direction change to happen. So that we found it to be, though it's expected, it goes to show that the complex interplay between the dynamics of flagella and cell body, in particular to the related positions of the flagellar attachments, could be influencing the duration of tumbling. Okay, I think I'm pressed for time here. I'll go to the next slide. So future directions involve studying the phenomenon of wall entrapment, as you can see in the right side GIF. This is an excellent experimental study from holographic microscopy. That will be the next step, because that's the first step in bacterial film formation. So as well as we would like to study the same sort of wall entrapment and mechanism near wall in the presence of shear flows, because in the actual situation of the human body and all that, there are shear flows, there are like some fluid moving in the background. So that's about my presentation. And we are doing this in collaboration with Dr. Holger Stark of TU Berlin.
Okay.
These are my references. You can make note of the references as well. Thank you very much. Yeah.
Excellent. Thanks, Vignesh. So our next speaker is Chandan Kumar, who will be speaking with us about effect of monovalent salt on lipid nanoclay composite layer at the air-water interface. Chandan?
Yes, sir.
Yeah. So you may stop sharing, Vignesh, and Chandan, you can start sharing.
Okay. So one second. How do we stop sharing?
There is a red button, right? Like stop share. You see that
your
screen.
Yeah. I see it. Yeah.
Okay.
Thank you.
Chandan, please start.
Are you able to see my screen?
Yes.
I'll start with thanking the organizers to allow me to present a part of my ongoing postdoctoral research work, which I have been doing at Indian Institute of IIT Bombay under the guidance of Sunita Srivastava. So, as the title suggests, I have been studying a role of monovalent salt on stability of lipid nanoclay composite at the air-water interface. So before going into my actual system, let me introduce this lipid molecule, and its functions. As we know, this most part of human body is lipids are very basic building block of living cells, and it plays important role uh, in giving signal, preserve energy, transmit signals, and maintain temperature within body. And these lipid molecules are dynamic and viscoelastic in nature. Its intermolecular interactions decide its mechanical property. Lipids and these lipids molecules are sensitive to temperature, pH, and ionic strength of the media. As the lipid molecules are amphiphilic in nature, that means it has both hydrophilic and hydrophobic part attached to it, and because of this its biocompatibility, it is majorly used in pharmaceutical sector. So before let me introduce this particular slide is for those those who don't know what exactly language velocity is. So what we do in our lab is we take composition of DM tap and DMPC. DMPC is a Jupiter ionic molecule, and DM tap is a cationic lipid, and we mix these two particular lipid in various composition. That's how we have we have flexibility to create an interface of no surface ion to 100% surface ion, and we inject this lipid leponite molecule, which is a discotic molecule with an ionic charge present at its surface, and at edge it is positively charged. And over time, this reaction happens between this cationic lipid and this leponite molecules, and that's how, and the interface looks something like this. So so what we do is we vary this salt concentration of the surface medium at which we spread this monolayer, uh, and we take isotherms. Isotherm means these experiments were performed at particular temperature, and that is at room temperature. So as we vary salt concentration for a 10% of cationic cationic lipid. So we see with increase in salt concentration, we have when there was no leponite present in the surface, we do see the area change in area related to its original area changes with respect to as we increase concentration of salt. Whereas in a medium where leponite was injected in the medium, we hardly see means the change is very minimal. So talking about its stability, this stability is represented as a function of elastic modulus. So as you can see, as we increase salt concentration, the modulus increases, and when there was no in a leponite free medium, with respect to increase in the salt concentration, what we do see is the modulus increases monotonously, and with respect to no salt concentration, and with 100% of salt concentration, 100 millimolar salt concentration in the surface gives almost twice a higher modulus of this. That means the stability is increasing with respect as we are increasing the salt concentration.
Chandan, you have about a minute to wrap up.
Yes, sir. So last slide. And in the similar kind of stability we could achieve with 5% of 5 millimolar of salt concentration with leponite in the surface. So that this on basis of this one, when we conclude that surface ion presence of surface ion plays means it helps the lipid molecule to condense in highly ordered state. So based on that one, we here we compared two different in a salt free state free medium. What we see with increase in surface concentration of cationic lipid, the absorption of leponite increases, whereas when we inject in a salt in a 5 millimolar subphasic concentration, we with increase in the the physiosorption is stopped, means it get hindered because of this subphasic ion. So that on basis of these experiment, what we concluded that physiosorption is a direct function of ionic concentration at the interface, whereas it is inversely related to the ionic concentration in the surface. Thank you from my side.
Okay, thank you. Excellent. So now the floor is open for the presentations of Vignesh and of Chandan. Anybody has questions? Please open your mic and maybe you can just ask whomever you have questions. Okay, so in the meantime, I may ask a question to Chandan. Chandan?
Yes, sir.
So, when you're looking at this, the elastic modulus things that you're extracting from your Langmuir isotherms, there are two peaks, right? I mean, what would be the physical reasons for that?
These
How do we interpret
phases? This actually this near to this is near to collapse. Actually, near to collapse, the molecules start behaving randomly. As you can see in no salt concentration, also the black curve, which is I don't know, you are able to see on.
Yeah, we can see.
So this although the molecule collapsed here at near about 80 angstrom square, but because beyond this point, this the lipid molecule start forming multi-layers, and multi-layers modulus may be sometime higher than this individual.
But isn't for your black curve, for your black curve, the collapse transition is at much higher, I mean, much lower areas, right? Much higher compression. So the peak that you are pointing out right now, so that is not the collapse transition, isn't it?
This is actually collapse, sir.
Okay, so any other questions to Chandan or Vignesh? So Vignesh, are you? Yeah.
Yes, sir, yeah.
So I'm just like thinking about your future work, and which is excellent. So you want to look at the tumbling frequency of your bacteria like under shear. Is that what you're trying?
Yeah, that would be yeah. I mean actually tumbling. The tumbling, actually, it's not very clear whether the tumbling actually helps the wall entrapment or not. Actually, shear is needed to make it realistic. A wall entrapment doesn't happen in a vacuum. In the actual situation, shear flow is there anyway to make it realistic. We're going step by step to make it realistic. So that would be the more final stages. So tumbling frequency, I think you were asking about the sort of the Rio taxes or something like that. Whether the hydrodynamic interaction.
Yes.
That could be one thing that we could look at. But we could also look at the how the polymorphism and the hydrodynamic interaction, as well as state interaction upon landing or upon collision. So how that influences the overall motion, that also we could look at. There are a number of things we could look at once we come to the stage of detail in the simulation.
Okay, excellent. So anyway, we can discuss this with Tapan at some later point. So I think we can start the next set of people for the mini talk. So the next speaker is Hema, and she will be presenting about ion transport mechanisms in biocompatible lithium ion battery electrolytes. Is Hema here?
Yes, sir.
Yeah, could you please start sharing and then start your presentation?
Yes, I'm sharing. Is my screen visible, sir?
Yeah, please go ahead.
Good afternoon, everyone. I'm Hema from the Department of Physics, IIT Jodhpur. Today, I'm going, first of all, I would like to thank to organize to give me a opportunity to present here. I'm going to tell electrolyte which are using in battery electrolyte. So I'm investing in new electrolyte consisting of pectine, which is biopolymer octane. In many experimental studies, people propose this pectine, but there is no theoretical study found. And why pectine is used? Because pectine has a biodegradable property, and also it has a good mechanical properties and also increase the viscosity. So I would propose this pectine, and so in using MD simulation, we study this ion transfer properties in electrolyte with the different weight percentage of pectine in ethylene carbonate with the dissolved lithium TFSI. And what are the approach to do this is first we follow this equilibration protocol. First minimization and then NVT and then NVT at 425 Kelvin for a 10 nanosecond. After that, I started this production run for 100 nanosecond. And we have used this OPLS phosphate. This is optimized potential for liquid simulation. In my snapshot, we see that this red color is an pectine, and rest of them is ethylene carbonate, lithium, and TFSI. And as we discussed that we study this ion transport property, so transport of ions in simulation is calculated by this diffusion coefficient, and this diffusion coefficient is a function of mean square displacement. Using this formula, we calculated this mean square displacement. In my graph, this is MST versus time for lithium. In this graph, we see that MST is decreasing with the increasing weight percentage of pectine. And in the second, we see that MST for lithium and TFSI. For TFSI, MSD is higher than the lithium. And the diffusion versus weight percent of pectine, we also see that the diffusion coefficient is higher than the lithium. And we also see that up to 3 weight percentage of pectine, this diffusion coefficient is not changing so much. And after that, it is slightly increased. So how it is so that up to we see that this up to 3 weight percentage, there is no change in diffusion coefficient. So we study this relaxation phenomena from this ion pair correlation function. Using this formula, we calculated this ion pair correlation functions. And by fitting this ion pair correlation function, we calculated this relaxation time. The mathematically formula of this ion pair correlation function is shown this figure. In this figure, this tau versus weight percent of pectine, we see that this tau is increasing with the increasing weight percent of pectine. And in last slide, I shown that diffusion coefficient is decreasing with the weight percent of pectine. From these two graphs, we can say that this D is inversely proportional to tau, but is not exactly that is this inversely proportional to tau. Then like in D versus tau graph, we see that our beta is like degree of stretching is a 2.4 something. So from this we can say that D and tau is not simply directly correlated, and there is something from this interaction of the pectine, and this how's the interaction of pectine oxygen atom with the lithium ion. We studied this ion coordination with pectine, that how this lithium ion associate with number of pectine, and it increases with the increasing weight percentage of pectine, like for 50 weight percentage. And in from this
Emma,
conclusion,
you have a minute to wrap up.
okay, sir. Thank you, sir.
Good.
Conclusion is like we simulated this new biocompatible polymer electrolyte for lithium battery applications, and this we compared this pure EC lithium TFSI and pure pectin lithium TFSI electrolyte. Also, we discussed that there's a small amount of pectin, like up to 3 weight percentage. This does not seem to affect this ion transport properties. From these, we completely understood this ion transport properties and structural relaxation. And after that, I started this mechanical strength to the battery calculations. Like primarily, this my result is as shown in stress versus strain. But I started for only 3 weight percentage, and I'm also doing the this work on this. So in the future, I will be happy to say this better understanding, and also this mechanical property if I go started the working. So thank you so much.
Thank you. And so let's continue with the next speaker. The next speaker is Megha Khokar, and she will be speaking about tailoring the structure-induced resonances in soft material-based low dielectric metasurfaces. Is Megha here? Megha? I don't see her. Okay, it looks like Megha is not here. So I think then we can discuss the presentation of Hema. Anyone has questions for Hema? Questions?
Hi, can I ask a question?
Yes, please.
Yes, please.
Okay. So, Hema, in your second slide, it's just a question about equilibration, minimizing your system.
Yes.
Yeah. Can you go to, yeah. So, you are preferring NVT and then NPT.
Yes.
So, the choice of considering NVT prior to NPT, why is it so? Like, why do you choose NPT first? And then
Like first NVT is for this equilibrate our temperature, and then for the NVT, then and for equilibration for the temperature. Then I follow this path. Like first I equilibrate our temperature, and then pressure, then NVT, and then NPT.
But and through NPT also you can equilibrate, like you can go to a minima, like minima temperature. I'm always confused about this priority of NVT upon NPT in small scale project. For example, your project, the simulation or the size, I think, is not big. It is not big. So preferring NPT first and NVT afterward won't affect, I think, like, I don't know, you can like tell me. It won't affect the minimization process. Like, what is the condition you are considering that your system is minimized?
As you this thermodynamic parameters like temperature, pressure, we see that all these parameters.
Yeah.
And after the NPT, like if we our equilibrated our system, maybe some fluctuations or maybe some errors. So we extended this run for 100 nanoseconds. So after
that was
we
I'm
decided.
asking that how do you choose like this, like after how do you decide that this is my step done for NVT and I will go to NPT, the transition? Like, do you understand what I mean to say?
Yeah,
that,
I understand, but I don't know that exactly. So
like in the Longevin system, we know it's very, like, it is a bit complicated system. It's more complicated, I think. It's not a simple Longevin chain or Longevin, because there is a lot of I can see there's a lot of potential involved in this project. So yeah, that was my doubt, like how do you choose it, NPT or NVT or NPT first or NVT, and why can't you only go with NPT? Because since the system size is very small, I don't think so NVT is required, or it is. I still have.
I don't know the exact answer.
Okay. Thank you, Hema.
Okay, thank you. Any other questions? Okay, Hema, could you please go to the next slide? Yeah. So here, if I look at the MSD plots, like with respect to your weight percentage of pectin, or even the MSD plots with TFSI ion and lithium ion, so
Yes, sir.
you clearly change the behavior of your diffusion, right? Like, so you're going from diffusive behavior to subdiffusive behavior.
Yes, sir.
And then how do you extract diffusion coefficient for all these cases?
From the MST, sir.
How?
Sir, there's like we this Einstein relations, like first this calculated the slope and this using this Einstein.
Right, but that is only valid for diffusive systems, right?
Yes.
When you have systems which are not exactly diffusive, like sub-diffusive or super diffusive, we cannot just use a slope to get the diffusivity, right?
Yes, sir, but I just calculated in this way.
Because your linear relation doesn't hold any longer, right? So your delta square has to scale with t, then only you can get this prefactor d, right? And if you're not linear, that is your mean square displacement is not scaling with t, so then you cannot use that. Okay, so there's something for you to maybe think over.
Yes.
Any other questions?
I have a quick one.
Yes, Danny, please.
So if you can go to the next slide, or maybe the next one, the final one. Yeah. So what does the 50 weight percent mean? Like you have 50 weight percent of pectin, is it?
Pectin and 50 weight percentage of ethylene carbonate. So 50 weight percentage.
It looks like your stress to strain changes considerably, but why 50%? I thought this was an additive.
As I just started, like I don't say exactly, but I started this mechanical results. This started. I don't conclude that this maybe
Okay.
I'm going the right path or not.
Right, but from the perspective of the application, does it make sense to add 50% of an additive? I just want to know. I don't know. Does it make sense at all to have 50% of pectin for that application?
For 50% of pectin, can you repeat?
So adding an additive of 50%, does that make sense from the perspective of the application for which this lithium thing is used, the battery applications, right?
As we see that the pectin is maybe increase this mechanical strength. So we increasing this up to 50 weight percentage. So,
but then you are also losing in terms of your conduction, right?
This we won't simultaneously increase this conductivity and this mechanical strength also.
Which is exactly what you want to do with your additive, right? Like you want to use an additive to increase the mechanical strength. At the same time, you are not losing the efficiency of your battery.
That's right. So maybe you should look at that as well. Both, what is the advantage in mechanical strength you get at the compromise of the conductivity? Maybe you should look at measures like that to get that.
Yeah.
Okay, any other questions? Okay, if there are none, I think we can close the session, and I thank all the speakers and all for the interaction. So I hand it over to the organizers.
So we meet back at 1:45, right, Sunita?